Welcome to Chapter 10, covering muscle physiology. As you can see the learning outcomes here, please make sure that you are able to discuss and answer these questions by the end of the lecture. And just a few more learning outcomes here. Let's begin our conversation today talking about the general functions of muscle. Now, the majority of our lecture today is going to be skeletal muscle. That's going to be our main focus. We just got done covering the bones and the joints, right? We will discuss the other two types of muscle, cardiac and smooth, towards the end of the chapter. However, we're going to cover the majority of cardiac and smooth muscle topics next semester in anatomy and physiology too. We're going to have a whole chapter on the heart, and we'll have several chapters dealing with things or organs that have smooth muscle, right? Such as our respiratory organs, our digestive organs, our vessels, our urinary organs, those kind of things. So once again, majority of our conversation is skeletal muscle. But regardless, if we're talking cardiac, smooth, or skeletal, the main job for most muscle is to move. Your heart beats every single second. That's your cardiac. Smooth muscle, think about the hollow organs and your digestive system. Your esophagus, your stomach, your small intestines. They have to contract in order to move food from A to B, wherever it's going. And then, of course, our skeletal muscles have to move in order to move our bones and our joints, right? So we will be talking about how we move in great detail. You're going to be shocked of how many crazy things have to happen within milliseconds in order for you to move your finger one time or blink your eyelid. Something that seems so simplistic that we take for granted is extremely complicated. But you'll get to learn it very shortly. Another big function of muscles is heat production. We generally give off a lot of heat when we use our muscles, right? If you don't believe me, go run around your place 2 times real quick. Crank out 10 push-ups or do some jumping jacks, and your body will give off heat. You might even start sweating. The last part, or the last big function for skeletal muscle, is posture. And this is something that we don't always think of throughout the day. We usually think of exercising or working out when using our skeletal muscles, but we use postural muscles all the time, every day. So right now I'm standing, and there's just hundreds and hundreds of muscles that I'm using. All the muscles around my feet and toes and ankle, my calf muscles, my hamstrings, my quads, my hip, glute muscles are working. Also, my abdominal and lower back muscles are working to keep me upright and balanced. If you are sitting right now, same thing. Maybe your abdominal muscles and your lower back muscles are keeping you upright. If you're lying down, you got to probably keep your head up. There are postural muscles in your neck to make sure that your head just doesn't flop over. So posture also going to be a big thing that skeletal muscles do, and we'll have a specific type of contraction that we'll introduce down the road from here. But these are the 3 main functions of our muscles. Moving on to the characteristics of skeletal muscle, 2 very important bullet points on this slide. Let's tackle the first one. First, skeletal muscle tissue is composed of muscle fibers, aka cells. So you're going to hear me do this a lot on purpose. You're also going to hear me do this on accident. A muscle cell is the exact same thing as a muscle fiber. So if somebody says you have fast twitch muscle fibers or you have slow twitch muscle fibers, they're saying you have fast or slow twitch muscle cells. Okay, they are the exact same thing. So let's take a look at this picture on the right and try to dive in and figure out what in the world's going on here. So on the top picture, what you have, skeletal muscle, this whole big thing, this red thing, I'm circling the entire structure. That is our muscle tissue. Now notice there's this kind of white cord that's attaching to the muscle, and then that's going to attach to some type of bone. So remember, the structure that attaches muscle to bone is called a tendon, right? And then ligaments are more for bone-to-bone connections. So we have a tendon connecting our entire muscle to a bone. All right, all of these structures in our muscles are organized very specifically. So we have 3 layers of organization, and around those 3 layers, we have 3 protective coatings, mainly made up of collagen fibers, which should make sense. We talked about collagen a lot so far this semester, and what we're trying to do here is protect the muscle and protect the things inside the muscle. So what type of protein fiber would be really helpful in protecting things? You got it, collagen. So, surrounding the muscle, and what I'm going to do is I'm going to highlight it in black. Surrounding the actual muscle itself is a protective coating, once again, of collagen fibers, and what that's called is the epimysium. And you can see the spelling right here. Some people pronounce it epimysium. That's okay too. They're both are commonly accepted in the anatomical world, if you will. I was always taught epimysium. So once again, the epimysium, all it is a bunch of collagen fibers protecting that muscle. But it's protecting the entire outer portion of the muscle, the superficial aspect of the entire muscle. So epi, if you just go back to our prefixes from, we probably covered this around chapter 3 or 4, means over or above. If you remember the epidermis, that's a great example. The epidermis is the layer of skin above the dermis. So that's what we're talking about here. The epimysium is kind of above or on top of the entire muscle. All right, so let's clear that out and take a look at what's inside of the muscle. And what you'll see are these tiny islands, or these little colonies throughout the muscle itself. So what I'm doing is I'm circling them currently. And what these islands, or these colonies, are called are fascicles. And you have dozens and dozens of fascicles inside of a muscle. We're just showing you a few here. So surrounding the fascicles is also another layer of collagen fibers trying to protect it, and what that's called is the perimysium. And you can see the spelling right here, P-E-R-I, perimysium. And peri, kind of similar to epi, peri just means around. So this is kind of, if you will, the middle layer of the muscle tissue. We're going to have one more in just a second. So the epimysium protects the entire muscle, and the perimysium is what's protecting these fascicles. And then, if you actually look inside of the fascicles, so for example, what we're doing, where I'm drawing the arrow right here, this is one big fascicle that we're kind of drawing out a little bit. And then that's what we're doing here. We're zooming in on this second picture where I just drew the arrow. So this right here is not a muscle. This is just one fascicle. And inside of the fascicles are individual muscle fibers. And once again, muscle fibers are also known as muscle cells. So I'm going to draw some numbers, or write some numbers. 1, 2, 3, 4, and then this will be 5. These are all different muscle cells. So several cells are wrapped inside of a fascicle. And then last but not least, a muscle fiber or a muscle cell also has a protective coating around it. Each individual muscle cell has its own little coating, tiny little collagen fibers protecting it, and that's going to be called the endomysium. And endo means inner, inside, or within, right? We learned about endocytosis, bringing something into the cell. Same thing here. Inside, deep inside of the muscle, we have our muscle cells, and protecting that is called the endomysium. And you can see a picture right here with the individual muscle fiber, and you can kind of see this little coating being peeled off. and that would be the endomysium. Now, if you're jumping ahead, if you're looking at this bottom picture and you're seeing, well, that's one muscle cell, and then you have all these crazy structures inside of that, well, yeah, it gets even more complicated. We're going to get into that in just a sec. But muscles are made up of colonies called fascicles, and fascicles are made up of a bunch of muscle cells. And then we have the epimysium, the perimysium, and the endomysium, protecting those respectively. Okay, so that's the first bullet point. Second bullet point is skeletal muscles are able to do some really, really cool things that not all other tissues are able to do. And you can see these 3 bullet points. You have excitability, aka irritability. You have contractility, and you have extensibility. And we're going to dive into each one of these 3 on the next few slides. So the first characteristic is excitability. And you can see the other name for that is irritability. And all that means is the capability of a muscle cell being excited by a nerve cell. So you can take a look at this picture on the right, and on very basic diagram, but this big yellow thing, number 1 and number 2, what this is the end of a nerve. And even though we haven't studied neurology quite yet, we know that nerves carry electricity or electric currents, and we can actually take that electric current and shock the muscle cell or the muscle itself. And when the muscle is shocked, it actually contracts, which will be our next slide, our next bullet point. So it is able to be excited or irritated, right? If I shock you with electricity, you're probably going to be kind of irritable, right? So that's where they get that terminology from. But not all cells or not all tissues can do this. For example, say that we take a little taser and we shock just the edge of your skin, just the epidermis. Well, 1, a lot of your cells in the epidermis are dead, but 2, you don't have a lot of motor function in that area. You have receptors that can detect pain, right, just underneath that epidermis area, but your skin's not going to start moving or crinkling or get goosebumps or anything if you shock it with electricity. There's no movement, there's no response to that other than just the pain fibers going to your brain. So when you shock a muscle with electricity, it will respond, it will contract. And if you think about it, if somebody's having a heart attack and they need to re-shock them back into rhythm, that's what they do, right? They put the pads on, clear, and then they shock them. And that's to get the heart back in to a sinus rhythm. So excitability, irritability is the ability for nerves to talk to your muscles. Next topic is contractility, and this kind of goes hand in hand with the excitability or irritability. If you irritate or excite a muscle, it generally will contract. So you can see the effect of contracting is we are able to move our bones by pulling on those tendons, and then the tendons pull on the bones, and when you move your bones, you can move all those joints that we just learned. And the last big important topic is extensibility. So muscles are great at contracting, yes, but they also have to stretch a little bit. In order for the muscles to contract optimally or maximally, they do have to have a little elasticity to them, if you will, or what we call extensibility. So picture this, if you're trying to jump up on a chair, what you have to do is you have to stretch the muscle a little bit before actually contracting, right? So if I'm going to contract all my quads and my hamstrings and my leg muscles to jump up on a chair, first what I have to do is I have to bend my knees, bend my hips, and I gotta have to stretch the muscles, and then I can explode up and jump on the chair. So this plays a big role in helping the muscle contract optimally, but also it helps the muscles return to their original position. So we'll talk about why we don't want our muscles too loosey-goosey. Yoga and stretching is awesome, it's great, but too much of it, unless you're genetically predetermined to be really, really good at that, too much of that can be bad. We kind of want the sweet spot regarding our skeletal muscle cells. So we'll talk more about this later on. So just quick recap, 3 big characteristics of muscle. They are able to be excited or irritated by the nervous system. They're able to contract, and then they have this extensibility, this ability to stretch. For the next 2 slides, we're going to be talking about the structures that make up our muscle fibers, aka our muscle cells. And a lot of them are very similar to what we've already talked about in Chapter 3 with the cell. We're going to introduce the mitochondria and the endoplasmic reticulum and the nuclei. But because muscle cells are very unique compared to other cells, we're going to have unique names to a lot of these structures. So let's start with the first one. You can see in this bullet point is called the sarcolemma. And what the sarcolemma stands for, what it means, sarco means flesh, and lemma is something, it's like a sheath, or aka an outer covering or casing. And if you think about it, that's what the plasma membrane really is, right? The plasma membrane of the cell is an outer covering or casing around your cells. So we just have a special or specific name for the plasma membrane of a muscle cell. So instead of saying plasma membrane of a muscle cell, we just say sarcolemma. So sarco, flesh, meaning we're talking about, right, if you cut open my skin, if you go deep enough, you'll hit flesh, you'll hit my muscle, and then lemma is outer casing, outer covering. So sarco, you're going to see several times. The sarcoplasm should make sense. That's simply the cytoplasm of a muscle cell. And then last but not least, you have the sarcoplasmic reticulum, and that should sound kind of familiar, right? We talked about the rough endoplasmic reticulum and the smooth endoplasmic reticulum in Chapter 3. And that's what we're talking about here. Instead of saying endoplasmic reticulum of skeletal muscle, we're just going to say sarcoplasmic reticulum. And this is specifically talking about the smooth ER, not so much the rough ER. And although this seems kind of minor and not that big of a detail to highlight right now, this is going to be gigantic very, very shortly. The main function, the main purpose of the sarcoplasmic reticulum is to store calcium. Store calcium. Very, very important. If you go back to Chapter 6, when we talked about skeletal tissue, and we talked about the homeostatic regulation, the negative feedback loop with calcium. And we said we were talking about calcitonin from the thyroid gland, and we were talking about parathyroid hormone from the parathyroid gland. And those two are kind of always talking with one another, or kind of counterbalancing one another to make sure our blood levels of calcium are within homeostatic range. And we said that was vitally, vitally important. We don't like to get rid of calcium. We don't sweat it out. We don't generally urinate it out, and we don't defecate it out. We have to keep it in our body. It's too important to get rid of. So if we have too much calcium in the blood, that's a problem. That's going to cause all sorts of, we talked about different organs shutting down. We also talked about muscle issues, involuntary muscle contractions, or muscle paralysis, depending on if your calcium's too high or too low. Well, one very, very important aspect of muscle tissue is that it has to have calcium inside of the muscle cells themselves in order to make the muscles contract. So yes, calcium is important for your bones. It's also important for platelets and when clotting. It's also important for muscle contraction. And we're going to get to that in just a little bit. So to recap, we have the sarcolemma, which is the outer covering of the skeletal muscle cell. We have the sarcoplasm, which we're going to have everything else floating around, right? Your ribosomes, your mitochondria, all that crazy stuff we learned about. And then the sarcoplasmic reticulum is the smooth ER. Main job, main function, store calcium. Because once we release that calcium, we're going to help the muscle actually contract down the road. Other structures that are found inside of your muscle cells. First is mitochondria. And remember, we call the mitochondria the powerhouse of the cell or the energy factories of the cell. This is where we're going to be taking our breakdowns of mainly sugar, glucose, and combining it with the help of oxygen, and we're going to be making a lot of ATP. Now you notice in bold, it says muscle cells need a lot of ATP. So typically we have a lot of mitochondria in our skeletal muscle cells. And this is the really cool part about exercise physiology. You can actually develop more mitochondria in your muscle cells. So it's kind of perfect timing. I don't mean to toot my own horn or anything, but I never hated running. I never liked running long distances. I was always a real short sprint kind of guy, and just based on me getting older and dealing with injuries, I've learned that I can't really do that all the time, so I have to start running longer distances to maintain my aerobic fitness. So just a few weeks ago, I ran, I don't know, 1.5 miles, 2 miles at a certain amount of time, and that was brutal. That was really hard. I was sore for days and did not want to do it again. Fast forward 2 weeks, I ran 2.5 miles at an even faster pace, and I'm not even sore the next day anymore. And a big part of that is mental. You know, your brain has to be able to push you over that limit, but a lot of it is your body responds to exercise. And the more you do something, the more you push your body in a healthy way, you're going to adapt to that exercise. And one way your body does that is you make more mitochondria. So I struggled with running 1.5 miles, and now I can run 2.5 miles with less discomfort, and it's because my body is able to make more energy in those muscle cells, keep me going longer. Now, will I keep going 3, 4, 5, 6? No, I think I'm going to cap it there, but it was just a good example to show you that your body develops mitochondria based on your body's needs. So in this sense, no pain, no gain kind of does apply. Not always, but with mitochondria, it does. Moving along to nuclei. You have in skeletal muscle a very unique characteristic that in skeletal muscle alone, we have many nuclei per muscle fiber. So this is what we called multinucleation back in the tissues chapter. Remember, cardiac and smooth are a little more normal, simplistic, one cell, one nucleus. Skeletal muscle is the only type of tissue that has many nuclei per cell. And you can, if you look at the picture in the very top right corner, you can see with these red arrows that I'm drawing, you can see that there are multiple nuclei or connected or inside involved with one muscle cell. And the main reason we do this is, the main reason why we have this, is because of exercise. Generally speaking, your skeletal muscle cells do have one nucleus per cell as they're forming, but what we do at the gym and when we run or when we do with swimming, whatever, is we rip those muscles apart. And that's why we're sore the next day and a couple days afterwards sometimes. But when you sleep and when you have proper nutrition, those muscle cells heal and those broken muscle cells fuse together to form bigger, stronger muscle cells. However, those nuclei don't go away. So in this case, in the picture, this muscle cell is a byproduct of 3 original muscle cells, and you've torn and ripped them, and they've eventually fused together to form this big, much stronger muscle cell. So another good purpose of working out is not only do your muscle cells develop more mitochondria, but they can actually get bigger. And if your muscle cells get bigger, your muscles can get bigger as well. Also, that sarcoplasm can get bigger. All right, these last 2 bullet points we have not talked about. These are brand new terms, so I want to take your attention to the middle picture. And in the middle picture, what you'll see is something called a myofibril. So this cylinder-looking thing is called a myofibril, and myo means muscle. So whenever you see the word sarco or myo, we're generally going to be referring to muscle. And fibril means little fiber. So this is a little fiber inside of a muscle fiber. Let me repeat that again. this is a little fiber inside of a muscle fiber, or in other words, a little tiny thing inside of a muscle cell. So looking at the picture in the top right corner, this is one muscle cell. And then once again, you can see all of these cylinder-looking structures inside of the muscle cells. And what those are myofibrils. So many, many little fibers are stacked and grouped inside of muscle fibers. So within the muscle cells, kind of lying on top of these myofibrils, is our sarcoplasmic reticulum. And you see this kind of yellow orangey spider web looking structure also down here on this more zoomed in picture. This is the sarcoplasmic reticulum, also known as the smooth ER of your skeletal muscle cells. Now on the previous slide we said it's going to be very, very important, right, regarding that calcium. But for right now, let's just introduce these terms. So sarcoplasmic reticulum, spider web looking stuff, but this spider web looking stuff on the left side of the picture needs to be connected to this spider web looking stuff on the right side of the picture. Because what we're going to be doing is we're going to be sending electricity through the muscle cell. We're going to be exciting the muscle or irritating the muscle in order for the muscle cell to contract. So we need this kind of network. We need this avenue in order to accomplish this. And how we accomplish this is through these structures called T-tubules. All right. So we are trying to contract a muscle. So let's clear some of this out. And what I'm going to do is I'm going to draw a nerve. And remember our anatomy of a nerve. We have a cell body, we have a couple dendrites, and then we got a really long one axon that kind of goes wherever it needs to go. All right. So what's going to happen is this. We are going to send electricity down that neuron to shock the sarcolemma. Now, when you excite your muscles, when your brain says, hey, biceps muscle, move, flex your elbow. So right now as y'all are sitting watching this video, flex your elbow for me, bend your elbows. Okay, it was really, really fast if you actually did it. There's not a delay. When your brain says move your elbow, you move it. You can do it very, very quickly. There's not a long process, thought out process here. And the reason why is because when we shock the muscle with electricity, that electricity spreads very quickly and very evenly throughout the entire muscle cell. So your biceps is a pretty large muscle. It actually originates, it starts in your shoulder area. It actually connects to part of that scapula that we talked about, and then it goes all the way down to your radius bone. So when you contract, when you bend your elbow, the entire muscle from your scapula all the way down to your radius is contracting at the same time. It's not one half is near your elbows contract and the other one's just hanging out. When you flex your biceps, your whole biceps flexes and moves your elbow. So that electricity has to spread evenly and quickly. And how we accomplish this is through these T-tubules. And what these are basically indentations or invaginations of the sarcolemma. And what they do is they connect the sarcoplasmic reticulum throughout the entire cell. So the way I always learn these is these are your big interstates. What we're trying to do is we're trying to go from our school to home, and say that we live 30 minutes away. So we got to get on the interstate to get there fast, right? So let's say that this speck right here I'm drawing in black, this little tiny speck, is where we live. In order to get there fast, we can take a whole bunch of back roads and get there in 45 minutes, or we can take the interstate and get there in 30, right? So what we like to do is we like to take the interstate and then just get off the proper exit. And then that exit will take us to our destination. So these interstates, these I-77s or 485s or wherever you live, these are the T-tubules. And what they do is they go all throughout the muscle cell from this kind of, in this picture, superior to inferior fashion, and they connect the sarcoplasmic reticulum to this electrical network. So once electricity is running down the T-tubules very quickly, it can spread throughout all parts of the sarcoplasmic reticulum. And when we get electricity spreading all throughout the muscle cell, we're going to get contraction eventually. All right, moving on. So now that we've covered the main structures in and around the cell and throughout the cell, such as the sarcolemma, sarcoplasmic reticulum, the mitochondria, the T-tubules, for these next few slides, we're just going to dive a little more into detail regarding the terms that we talked about, and we're going to introduce some new terms with the myofibrils and how what we're going to do is we're going to kind of just paint this picture of how muscles actually contract. So we're kind of just building on the knowledge that we've learned so far. So we've talked about the sarcoplasmic reticulum, and we've talked about the big interstates, the big highways, what we call the T-tubules. So this next slide is just introducing a term called the triad, and all the triad is just how specifically the T-tubules are connected to the sarcoplasmic reticulum. And to me, I mean, y'all correct me if you see something else, but it looks like a vanilla Oreo to me. What you see here is you see the T-tubules, which is this stuff in black, this frosting part that I'm coloring in black, and then you got the two golden crust, right? They call it a vanilla Oreo or golden Oreo or something like that. I don't know. Anyway, that's what a triad is. So you have the T-tubule in the middle, and then you have kind of the edges of the sarcoplasmic reticulum on either side, and what that's called is a terminal cisterna. And if you have more than one, which we do, we have two smothering or sandwiching the T-tubule, we call it cisternae. So what a triad is a T-tubule, and then you have a terminal cisterna on one side, and then you have another terminal cisterna on the other side. So once you get that, we'll just put a TT for T-tubule. So what you're looking at is three different structures. You got one terminal cisterna, a T-tubule, and a second terminal cisterna. And those three things together equal a triad. And once again, this is very important because this is what's going to allow those nervous impulses, aka the electrical signal, to spread very quickly throughout that sarcoplasmic reticulum. And once again, the reason why we want that sarcoplasmic reticulum being stimulated all at the same time is so our muscle cells can contract all at the same time, right? We don't want to delay in muscle contracting. We see that with really bad disorders such as multiple sclerosis, Parkinson's disease, and that's a lot of it, it's due to the nervous system not talking with the muscles effectively. So we want this structure, this anatomy, and it's called a triad. Once again, myofibrils are just little tiny fibers inside of a muscle cell. Now, by no means do you have to know this number, but you can see this picture doesn't quite do it justice. You can see in the picture there's maybe 15, 20 myofibrils, but there's over 1,000 per muscle cell, and you have thousands of muscle cells in your muscle. So you start multiplying and doing the math, it gets a little crazy. You have a lot of these structures. Now, what we're going to introduce in just a little bit is the myofibril is what's going to have the contractile proteins. And remember, a big characteristic of your muscle cells is the ability to contract. We call that contractility. So how do these muscle cells actually contract? Well, first you have to irritate it, right? You have to excite it with electricity. But then what? We have proteins, and these proteins actually move inside the myofibril, and they move closer together. And when they move closer together, the myofibril moves. And when the myofibril moves, the muscle fiber or the muscle cell moves. And when you get hundreds of muscle cells moving, then your muscle moves. So now what we're doing is we're kind of zooming in big time at the very, very smallest of microscopic proteins to see how this works, and then we'll zoom back out from a big picture and put it all together for a big old puzzle piece. Okay, a lot going on in this slide. What we're going to introduce here in this slide are the myofilaments. So there are 4 proteins that help with muscle contraction. 2 of them are what we call contractile proteins, which are actually doing the contracting. 2 of them are called regulatory proteins, which are actually doing the regulating of the contracting, making sure we don't contract too much or too little. Now, out of those 4 proteins, only one of them is what we call a thick protein, because it's just a lot bigger and thicker than the other 3 proteins, and therefore all the other ones are called thin. But what I want to do first is introduce the 2 contractile proteins. So first, the thick contractile protein is called myosin. And once again, when I'm saying contractile protein, this is going to help our muscle contract. We talked about the 3 things that muscles are really good at. It's excitability, aka irritability, contractility, contracting, and extensibility. This is where that is starting to come into play. Myosin helps the muscle contract and also extend back to its normal place. The other contractile protein is called actin. So actin, you can see, is considered a thin protein. It's a lot smaller. If you take a look at this picture in the lower right corner, you can see actin are these little kind of green circles. And then you can see the myosin are these thicker pink looking things that look like kind of Twizzlers. And then they got these 2 golf club head looking structures. So myosin's a little bigger, a little thicker. Also, it stains thicker and darker on the microscope or in the microscope. So speaking of that, let's introduce how these look underneath the microscope. So because myosin is very, very thick, it is a dark protein. And because actin is very, very thin, it's a lighter protein under the microscope. Now travel back with me to chapter 4 and your histology lab quiz, where you were studying all the different types of tissue, and you had to know the difference between skeletal muscle and smooth muscle and cardiac muscle. And the big difference was skeletal muscle was really, really organized, really put together well, had the multiple nuclei, and it had those stripes. So remember what those stripes were called? They were called striations. And then that's it. We didn't talk about it. We said, all right, those are striations. Let's move on. Now this is where it comes into play, hopefully making some light bulbs go off here. Striations, all they are just the alterations of, or altering patterns of these 2 proteins. So if you take a look at the picture in the upper right hand corner, what you'll see is this myosin actin just a little bit smaller. These purple structures right here, that is the myosin protein, and then the green structures, the green lines, are the actin proteins. And what you get in your muscle cells is you get this band, this collection where it's just myosin, and then you get this area where it's just actin, and another area where it's just actin. And that continues throughout the entire cell. So we're going to have another area where it's just all myosin in that purple. And then next to that, we're going to have another area where it's all actin proteins in green. So what you're getting is the striations, the stripes. You get a light protein and then a dark protein, and then a light protein and then a dark, and then light and dark and light, and it keeps going and going and going and going. And that's where you see those thousands and thousands of stripes or striations in the microscope. It's just myosin actin, myosin actin, myosin actin. So now we can be a little bit more specific. We don't have to say striations or stripes. We can actually say, oh, those are the 2 altering proteins that just overlap each other, myosin actin, myosin actin. Myosin being the darker one and the thicker one, actin being the lighter one and the skinnier or thinner one. Okay, moving right along. With this picture in the upper right-hand corner, I want to introduce the functional unit of muscle cells and therefore myofibrils. It's called a sarcomere. Remember, sarco means flesh, and a sarcomere, as I just mentioned, is the functional unit of muscle cells. And what that means is this is how your muscles actually contract. It's this unit. For example, the functional unit of your brain are the neurons. If you take all the neurons out of your brain, you're brain dead. Your brain doesn't work. The functional unit of your lungs are your air sacs. We'll learn that next semester. But if you don't have air sacs in your lungs, you can't breathe in air, right? And it's to this extreme. So yeah, you die without air sacs, you die without neurons. Your muscles die, or at least they don't work if you don't have these sarcomeres working. So when the sarcomeres contract, the myofibril contracts. And when the myofibril contracts, the muscle cell or muscle fiber contracts. And when you have a whole bunch of muscle cells contracting, the muscle contracts. So the sarcomere, all it is just where this actin and myosin are kind of interchanging and overlapping. And we have these two important landmarks called Z-lines, or Z-disc is a more common term nowadays. And you can see where the Z-discs are. It's just where this actin and other proteins kind of just make this weird zigzaggedy line where there is no myosin. So as I mentioned, one Z-disc or Z-line is the start line of the sarcomere, and then this Z-line or Z-disc would be the finish line. I'm just going to put fin for finish. So what's going to happen is myosin and actin are actually going to bind to one another and pull these Z-lines closer together towards that midline. And when these Z-lines or Z-discs get pulled closer together, the sarcomere gets smaller, or the sarcomere contracts. And once again, when the sarcomere contracts, everything else in the muscle is contracting. So you can picture, if I draw our three myosin bands here and here, and then let's draw some actin. Got our weird Z-disc here, ziggity, 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 and then actin. And then we have some myosin. So here is our another Z-disc in gold. When one Z-disc contracts, for example, when this one contracts with a star, this one kind of also has to contract because it's connected to this Z-disc. So one Z-disc, that's a starting line for a sarcomere. It's also a finish line for the adjacent sarcomere. So once these things start pulling together, they kind of get yanked towards the middle and they all start crunching up and squeezing. And that's how we get this muscle contraction started in relation to the myofilaments, these proteins. There's going to be a lot of steps that lead up to it in just a bit. All right, so almost done with this slide, and then we'll move on. How does myosin and actin bind together? How are these things working together to actually contract the sarcomere? Well, take a look at the anatomy. If you look at the bottom right pictures, again, you can see that the myosin has these two golf club looking head looking things, or balloon looking things. And these golf club heads fit really snugly in these holes inside of the actin. So what happens is myosin binds to those holes with actin, and then with the help of ATP actually moves it towards that midline, so the sarcomere, the start line and the finish line actually get closer together. But as long as we had ATP within the area, myosin and actin are kind of like boyfriend, girlfriend that just started dating. They're inseparable. They're attached at the hip. All they want to do is be with each other. Nobody else cares or matters in the world. It's just them two. So we need to control that, right? We need to make sure that they're eating and that they're doing their homework and doing their job and hanging out with other parts of their social world, their family members. Kind of a bad example, but you get what I'm getting at. So we need to regulate this. Because if you think about it, if myosin and actin are inseparable, if they're always attached to the hip, that means we're always contracting our muscles, which is not good, right? You don't want to contract your muscles 24/7. You're going to have cramp spasms, and you're not going to be able to straighten your joints, and we're going to have all sorts of pain and issues. So we need to regulate this. And this is where these two regulatory proteins come into play. And they are thin proteins, and their names are tropomyosin and troponin. Now, don't get these confused. You might have seen these words before. Tropomyosin is not myosin. It is different. It's a completely different protein. One's thick, one's thin. Myosin is a contractile protein, contracting. Tropomyosin regulates this. Totally different. Some of you may have heard of troponin, or some people pronounce it troponin, troponin, many different ways of saying it. In relation to heart tests, if somebody has a heart attack or some type of heart issue, they'll look at your troponin levels. Totally different. Same pronunciation, same spelling, completely different meaning here, completely different protein that we're talking about here. This has nothing to do with the heart in relation to this slide and to how your skeletal muscles contract. So let's talk about how these things actually work. How do they regulate myosin actin from hanging out all the time 24/7? So tropomyosin is this rope-looking structure that you see blocking, whoops, let me draw that arrow a little better, blocking the actual actin circles. It's that orange rope or that orange string. So if there's a big string in the way, myosin can't get inside of that circle. So they can't get together, and if they can't get together, they can't bind. They can't work. So our muscles are going to relax. Cool. That's great. Except for when we want to contract, then we have to move that rope out of the way, right? That's going to be important. So what in the world is troponin? Troponin is the protein that kind of controls the rope. That's kind of the brains of the situation. So you see this little kind of yellow gold nugget inside of the rope that I'm circling? That's troponin. And the way I always picture this in my mind, describe this to other students, is troponin is the gate or a lock. Well, yeah, troponin is the, excuse me, let's back up. Troponin is the lock or the keypad. Say that we're trying to break into a facility, or we're trying to get into a gated community or something, right? You need to unlock the keypad, and then the gate will open. All right, so the gate is tropomyosin. That's the rope that's in the way. All it does is move back and forth, open, close, open, close. But in order to get inside of that gate, you have to unlock the gate, or you have to unlock the keypad. And troponin is that keypad. So we have to distract the keypad, or unlock the lock, or distract the guard to open up the gate. Okay, last thing on this slide is, what is going to distract the guard, or what is going to actually be the key to our keyhole, or our key to our keypad? How are we going to get the gate open? Well, we have a very, very important element, a very important chemical that serves as our key to our keyhole, and it's stored in the sarcoplasmic reticulum. So if you go back a few slides in your notes, what did we say? What very important ion, very important electrolyte is found or stored inside the sarcoplasmic reticulum? And your answer should be calcium. Exactly. So calcium is what we're going to use to distract the guard or open up the keyhole, whatever analogy you want to use. Then we're going to move that tropomyosin rope or gate out of the way. And then myosin and actin can get together and make the sarcomeres contract, or those Z lines come closer together. So once again, emphasizing the importance of calcium. Calcium is going to help regulate your muscle contraction. So we contract our muscles when we want to, not when we don't want to. So let's move on and keep diving into more detail. You might want to pause and take a little breather after this slide. A lot of information. But once you're ready to keep going, let's move on and keep discussing this in more detail. So now that we've covered all that material, this slide just serves as a nice little review, putting it all together. You can see that in the top bullet point, actin and myosin are always attracted to one another. This is the new relationship, the boyfriend-girlfriend always wanting to be next to each other. As long as there's ATP present, they're always going to be trying to bind together. And what we call that bind, or that bond that's formed, is a cross bridge. So they kind of just link together or hold hands, and that's called the cross bridge. And I'm going to introduce that in more detail in just a little bit with some drawings, but that's myosin actin. Then we have our gate, or the actual thing inhibiting the myosin and actin from getting to one another, that rope-looking protein called the tropomyosin protein. And then the brains of the operation controlling the tropomyosin, or at least the one that's going to move tropomyosin out of the way, is the keypad, or the keyhole, or the security guard guarding the gate, whatever analogy you want to use, and that's what we call troponin. So these two, tropomyosin and troponin, the two ones that start with T, these are once again called regulatory proteins. They help regulate how much we contract. And then myosin actin, myosin being the thicker one, actin being the thinner one, these are the contractile proteins. And then we come to the sarcomere, and once again, the sarcomere is the functional unit of muscle contraction, also known as the contracting units. So the myofibrils and the muscle fibers don't contract unless the sarcomeres contract. And they're really just distinguished by these Z lines or Z discs. So if I was to draw these Z lines, if I was to draw 1, 2, 3, 4, 5 Z discs, you would know how many sarcomeres there are. You can see 1, 2, 3, 4 sarcomeres. Because remember, the Z disc just serves as the start line and the finish line. And what happens is when we contract our muscles, these Z discs get closer together. So this Z disc will kind of start moving towards that middle area right there. And this Z disc will move towards that middle area here. And they all just start getting crunched up. And that's what you can see in the picture on the right, where if you notice this top illustration, you can see I'm going to draw big red lines where the Z discs are supposed to be represented. So here's a Z disc. And then here's a Z disc. And this is during relaxation. And what the picture on the bottom is showing you is contraction. And you can see the Z discs are actually getting pulled closer together. And you can see how there's some overlap, or there's some, basically the picture is just closer together. The sarcomere is smaller. And that's what happens when contraction occurs. So now what we're going to do is we're going to zoom out and get the whole big picture, starting from the nervous system, starting from the brain, and then work all the way back to our cross bridge, or where these myosin actin actually connect to one another, bind to one another, and move the sarcomere. So this is just an introductory slide of where we're going to be going. For the next 3 to 4 slides, I'm going to be drawing for you, just to give you a kind of a step-by-step illustration of how this works. There's a lot of neat YouTube videos that put this in kind of a 3D format, and they show it in like a 2 to 3 minute window of all the different steps happening. I would strongly encourage you to check those out. Just YouTube and Google the titles of these next few slides. You can see here it's sliding filament theory near the middle of the page. The next one's going to be called the neuromuscular junction, and I'm going to show you everything you need to know. But if you're more of a visual learner and you want to see the moving parts and you just want to see better illustrations than my drawings, I would strongly encourage you to look at those. They will help connect all the dots potentially. So the picture on the left, this is where we're getting. The picture on the left and the picture on the right are actually the same thing. What we're looking at here is, in green we have the myosin, the thick protein, and then the actin are these little purple blueberry looking things. And then we have the blue rope that's blocking actin. So you can see here that myosin and actin cannot get together. They can't bind properly because the rope, the tropomyosin, is in the way. So what we need to do is we need to distract the security guard or unlock the keypad with the key. And you can see that the keypad is once again called troponin. And our key, or our big distractor, is that very important ion, calcium. So I'm gonna show you all of this again in my version, my drawings. But this is what we're working towards. This is the final product, because once that happens, once the calcium distracts the troponin and moves the tropomyosin out of the way, then what we get is we get the picture on the right, and we can see the sarcomere actually contract. So here in white are the two Z discs for the picture on the top, and then you can see the Z discs actually get closer together. So the picture on the right is showing contraction of the muscle on the bottom right. The picture on the top right is showing relaxation. So R for relaxation and C for contraction. So let's move on to the next few slides. They're going to take a little bit because it's going to take me a while to draw with this mouse, but it's going to show you step by step to how to move your finger one time or your toe one time, whatever we're moving. So I'd encourage you to pause the video and kind of copy what I'm writing, what I'm drawing, and then review it. And as you review it, you can fast forward it and make it as quickly as you want it to go. So let's get started. Okay, class, it is art time. So what we're going to do is, as I mentioned, we're going to start totally zoomed out. We're going to start all the way at the beginning. Anytime you move the muscle, it has to start in the brain or originate in the brain, or at least in the spinal cord. And then we're going to zoom in and zoom in some more and zoom in some more and learn all the moving parts for this, what I call a domino effect of muscle contraction. So I'm going to have 4 different pictures for you, and we're going to go in order with the 4 pictures. And if we're missing one part of any of these 4 pictures, this whole thing falls apart and the muscle doesn't move. So a lot of times when there's muscle paralysis, it can be due to one of these steps that we're going to talk about missing. Generally speaking, paralysis is a nervous system. This picture right here, the neuromuscular junction, but it can be something else as well with other disease processes. So what I'm about to draw is just a gross representation of the neuromuscular junction. The neuromuscular junction is also known as the NMJ. And all that means, it's very simple. Neuro is referring to the nervous system or a nerve cell, and muscle or muscular is muscle. So it's a nerve talking to a muscle, and a junction is just where 2 things meet or get together. So once again, characteristic of skeletal muscle is that it is irritable, it is excitable, it needs to be stimulated electrically in order to contract. So what I'm going to draw is our nervous system. Very plain Jane nervous system. So kind of what's in control of your nervous system is your brain, right? Here's your big brain. And then we have a brain stem attaching the brain to the spinal cord. I'm not going to draw the brain stem. I'm just going to draw a spinal cord going straight down. Okay, and then on the right side, I'm going to draw our muscle. And let's just say this is going to be well, for now, this will be a finger muscle. So M for muscle. Now, we haven't talked about neurology yet. We haven't talked about the divisions of the spinal cord or the lobes of the brain, but you actually already know all of that. You know the lobes of the brain are just protected by the bones of the skull. So you know there's a frontal lobe protected by the frontal bone. You know the parietal lobe is protected by the parietal bone. There's a temporal and occipital. You also know the divisions of the spinal cord. So in the spinal cord, we have these, what we call nerve roots that branch out from both sides, left to right, and they go all over the body. So some go to the left arm, some go to the right arm, some go to the left side of your face, others go to the right side of your face. Well, that's more of your brain, but still. Some go to the left toes, some go to the right toes, and they just go all the way down to the edge of the spinal cord. Now, the three divisions of the spinal cord are similar to the three divisions of the spine, the spinal column. You learn that the bones of your neck are called cervical bones. All right, so I'm going to put a big C in that neck region for cervical. You know the bones in the middle of your spine that kind of connect to, well, that do connect to your rib cage. Those are called thoracic bones. So we have a thoracic region of the spinal cord. And then your lower back was called the lumbar spine, and therefore we have lumbar nerves, lumbar nerve roots. So depending on what part of the body we're moving, what part of the body we're talking, we're going to have different regions of the spinal cord affected. So for example, with our muscle analogy here, I said finger. We're going to move a finger one time. So your finger is attached to your wrist, which is attached to your elbow, which is attached to your shoulder. Right, so this is an arm or an upper limb structure, and all upper limb structures are innervated or connected by nerves thanks to the cervical region. So the nerves in your neck go out to the nerves or go out to your arm structure. So if you ever have a pinched nerve in your neck, you may have some pain and numbness, tingling, shock sensations going down one of your arms, potentially. Depends on how bad the pinch is, or the herniation or bulge or whatever. But if we were talking about a lower leg muscle, let's say we're talking about our calf muscles to help move plantar and plantar flex, our ankle and foot area, right? If we were talking about a calf muscle, we wouldn't be talking about nerves in the cervical spine. We would be talking about nerves in the lumbar spine going out to the muscle. So it just depends on what we're moving here. So upper body, upper limbs are going to be innervated or connected. What that means is the nerve touches or connects to that area or gives nerve flow to that area. So upper body is going to be mainly cervical. Lower body is going to be mainly lumbar. And then that middle area, kind of your organs and rib muscles and that kind of thoracic region would be the thoracic spinal cord. That kind of makes sense, right? So this is all I want to draw on this page. What we're going to be doing is all we're going to show you is we're going to start the electrical message, or what we call an action potential. Once again, it has to start in the brain. And that electrical message is going to go down the brainstem, down the spinal cord, and it's going to go out the nerve. And it's eventually going to cause this whole cascade of effects on the muscle. But not yet. Not yet. So what I'm going to do on this next picture is I'm going to zoom in on this neuromuscular junction. So this purple circle that I'm drawing, we're going to zoom in on that relationship on the next slide. Now, one quick note though before we move on to the next slide is notice there is a tiny, tiny space between the neuron, the nerve cell, and the muscle. It's very, very small, but there is a tiny space. I'll draw it in purple right here. There is a space between the nervous tissue and the muscle. So when I zoom in, I'm going to draw a much bigger space to kind of show you a blown up view of that. So let's move on to the next slide and we'll talk about this. All right, everyone. So on this slide, we're going to once again zoom in on the neuromuscular junction where the nerve is right next to that muscle. And we're going to introduce a lot of moving parts and a lot of different steps here. So what I did prior to drawing the picture was I kind of gave us a little legend or label on the top of the slide. Excuse my handwriting. It's just really hard to do this with a laptop mouse, but that's actually pretty good for me. What I'm going to draw is a bunch of these different colors, and these colors are going to represent the different structures above. So anything in red is going to be the muscle. In black is going to be the neuron or the nerve cell. We're going to have some calcium-related things in blue, and you can kind of just read the rest, and I'll introduce those as we're drawing them. Okay, so let's start drawing some structures. We have once again, our big finger muscle. So M for muscle, and it's in red, so that's going to be our muscle for this picture. And then we have our big neuron. So the neuron kind of gets a little fatter, a little wider at the very, very end, where we're going to learn about the different parts of the neuron in the nervous system chapter, but that's our neuron in black. And then along the neurolemma, or the outside part, the plasma membrane of the nervous system tissue, the neuron in this case, are calcium channels. Now, we talked about channels before. We talked about different types of proteins, channel proteins and carrier proteins and that kind of thing. Basically, what these are proteins on the edge of the neuron that are not allowing calcium into the neuron. So they're blocking calcium, if you will. So we have this big concentration gradient. So remember, we talked about sodium and potassium in great length. We talked about how there's a lot of potassium on the inside of the cell, 10 times as much as sodium. But then on the outside of the cell, there's a lot of sodium and not a lot of potassium. So, and then we talked about the sodium potassium pump regulating all that. Here, with the nervous tissue, the neuron, we're going to be having a lot of calcium on the outside of the cell. So all those dots on the outside are trying to get in the neuron, right? This is a huge concentration gradient, which means there's a huge imbalance of ions. So we have, 40 calcium ions on the outside and 0 on the inside. So they want to get inside, but they can't. Those proteins are blocking them. And the only way that those proteins are going to get opened is if we shock them with electricity. So a really big theme in neurophysiology is if you shock something with electricity, a lot of times something happens, something changes shape or something moves. And that's what's going to happen with our muscle, as you'll see. All right, so we have calcium and the calcium channels blocking the calcium from getting in. Now, inside of this edge of the neuron, so the edge, or this part of the neuron that's talking to the muscle is called an axon. We talked about that before. And the very, very tip, or the very end part of the axon, is simply called the axon terminal. Just terminal meaning last step, right? So inside of this axon terminal, we have vesicles. And you know what vesicles are. They're just bubbles. We talked about specialized vesicles such as lysosomes and peroxisomes earlier in the semester. And inside of these vesicles, you have a chemical. And the chemical is, because it's found inside of a nerve cell, it's called a neurotransmitter. And that's what this NT in green stands for, neurotransmitter. So it's a chemical inside of a nerve. If we were talking about a chemical floating around in the bloodstream, we would say, oh, well, this is a hormone, right? But since this is only in the nerve, this is not a hormone, it's a neurotransmitter, nerve chemical that the nerve's going to release. Now, the name of this neurotransmitter, you do need to know. We're only going to talk about 2 or 3 different neurotransmitters this semester, and this is going to be the main one. 90% of our focus is going to be this. It's called acetylcholine, and you can see the spelling up here. It's A-C-E-T-Y-L-C-H-O-L-I-N-E, acetylcholine, and oftentimes we abbreviate that as ACH. So acetylcholine is the name of the neurotransmitter in our nerve cells going to our skeletal muscles. Other neurotransmitters that you may have heard of before, things like dopamine, serotonin, those kind of things, they're just chemicals that are released in the nervous system, mainly in your brain. But acetylcholine is what we're focused on right now. And then last but not least, we're going to have our acetylcholine receptors. And these are going to be found on something called the motor end plate, which is basically a wrinkly folded part of the sarcolemma. So on the very edge of the muscle plasma membrane, we have receptors for the acetylcholine. So you don't have to be a psychic to figure out what's going to happen. Eventually what we're going to do is we're going to get all that green acetylcholine out of the neuron, and it's going to bind somehow to these purple acetylcholine receptors. And then that is going to cause a whole nother thing to happen. So once again, this is a big snowball effect or a big domino effect. Once the first domino falls, once the brain says, hey, move my finger, all these steps are going to happen in succession, and eventually we're going to move our finger. But this happens in milliseconds. I'm drawing it really slow so we understand what's going on, but it's going to be happening pretty quickly. Okay, a couple more things I want to introduce, and then we'll see how this actually works. Once again, this whole picture, we have a neuron next to a muscle. This is called a neuromuscular junction. Another term for this is called a synapse. And a synapse is really just where a nerve talks to something. And in this case, it's a muscle, right? But it can be another nerve. It can be something like a sweat gland. It doesn't matter what. But if a nerve talks to something else, it's called a synapse. So this whole picture, I'll put in black temporarily, or purple, because I'm in purple. This is called a synapse. And since we're talking about muscle, we can also call this synapse the neuromuscular junction. It's pretty much, it's the same thing. Now the space between the neuron and the muscle, the space in this synapse, has a special name. It's called the synaptic cleft. And I'm probably going to run out of room, but synaptic cleft, meaning just it's a space. So all of this space right here is the synaptic cleft. And that is where our neurotransmitter is going to be secreted in just a second. So just a little extra vocabulary, because you're going to see a lot of this. You're going to see synapse and synaptic cleft later on in the chapter and in the nervous system chapter. So I want to introduce that now so it's not brand new. So let's erase that now that you have that down. Cleft and the synapse. Okay. So going back to our previous picture, remember it all starts in the brain. So there's actually going to be two electrical impulses that help us move that myosin actin, which help us move those Z discs, which helps us move that sarcomere. Two electrical activities or electrical processes. One, always going to start in the brain, always. Sorry, I say always. Like a reflex, every once in a while, it'll be the spinal cord. But if we're consciously moving our fingers and toes voluntarily, 100% it's going to be in the brain. The second one is going to happen in the muscle cell itself. So first is brain, second is muscle cell. But the reason why we have to have two electrical impulses is because look at the synaptic cleft. These things are not connected. And if they're not connected, electricity is disrupted. Right? So right now I'm sitting here on my computer, on my laptop, and I have it plugged into the wall because it's on low battery. But what if I took a pair of scissors and just cut the middle of that wire? But I put them right next to each other. There's a space with the cut wire, but they're right next to each other. Will that electricity from my outlet get to the laptop? And the answer is no. Even though they're really, really close together, if you break the circuit, you're breaking the electrical current. So it's not going to charge the laptop. So what we need to do is we need to have some type of intermediate or some type of spark to get a second potential in the laptop to charge it. And that's what we're going to do in just a moment is we are going to have the electrical impulse flowing from the brain down to the nerve. And then we have to release a chemical. And that chemical is going to be a neurotransmitter, that acetylcholine. And that chemical is going to start another electrical impulse, as we'll talk about. And one more thing I want to introduce to kind of help you with this whole process before we move on to the next slides are the order of ions that we're going to introduce here. So to help you with keeping everything straight in your head, we're going to introduce 3 ions in order of how this domino effect happens. And the order goes like this. It goes calcium, sodium, and then a different calcium. So calcium, sodium, calcium. Calcium, sodium, calcium. If you say that over and over in your head, you'll be able to answer any type of multiple choice matching question. As you can see in this picture, we have calcium. So this is the first calcium that we're going to introduce. And then that's going to lead to a sodium. introduction, and then that's going to lead to the calcium later in the sarcoplasmic reticulum. So calcium, sodium, calcium is how these ions are going to be introduced in this sequence. Okay, let's get to the show. So once again, the action potential or the electrical message, the nerve impulse is starting in the brain, going down the spinal cord, and then eventually it comes down the nerve. So in yellow, coming down is the big electrical impulse. And as we know with electricity is if you shock something, the electricity spreads very quickly, right? If you shock your finger, it's not just your finger that gets shocked with, like for example, if you get shocked with lightning on your hand's not going to be the only place where you have symptoms. Your whole body's going to get jolted depending on the amount of electricity, right? So this electricity spreads throughout the entire axon terminal. And once again, anytime you hit protein channels with electricity, a lot of times they change shape. And in this situation, what they're going to do is they're going to open. Now I'm going to try to clear these out without deleting my plasma membrane of my neuron, and so far so good. There we go. Awesome. All right, so what's going to happen is those protein channels are going to open, and what's going to travel in? So those protein channels were sensitive to calcium. They were keeping all the calcium out. So all of that calcium is going to flood the axon terminal or the nerve cell. So we're just going to have dozens and dozens and well, in reality, hundreds of calcium ions floating around in the axon terminal. Now, why did this happen? It's because we shocked it with electricity. Where's the electricity coming from? Well, it's coming from your brain. It's coming from the central nervous system. So calcium floods the cell. Then, once again, domino effect. So this happens, the electricity happens, then calcium comes in, and then calcium is going to cause something else to happen. So what calcium does is it causes the vesicles to move towards the synaptic cleft. It kind of forces them to migrate towards the edge of the cliff here. And then once they get to the very, very edge, what they do is they spit out the contents inside of the vesicle. And we learned about this in chapter 3. This is a form of active transport, and what happens is we have a vesicle, we have stuff in it, and whenever we spit something out of the cell, we call that exocytosis, right? Versus endocytosis would be bringing the bubbles into the cell. So in this situation, calcium causes those vesicles to move towards the cleft, towards the edge, and then they spit out, or they exocytose, all the acetylcholine into the cleft. And that's due to that calcium moving it there. Okay, so now we have a bunch of acetylcholine floating around in that synaptic cleft. And take a look what's on the other side of the cleft where that muscle is. We have receptors for acetylcholine. So then what's going to happen is simple diffusion. We have a boatload of acetylcholine molecules in the cleft and none binding towards the muscle. So then some of these receptors are going to be moving towards, some of the neurotransmitter is going to be moving towards the receptor. And then, similar to those calcium channels that got activated or open with electricity, whenever you bind a protein with whatever the receptor binds to, a lot of times that can change shape and that can open. So then what's going to happen is these acetylcholine receptors are going to open and allow another ion to come in. So this is going to be our second ion. Remember the order of our ions in this whole neuromuscular junction. We said calcium, sodium, calcium. So sodium is what's going to rush into the muscle cell. And remember back to our chemistry chapter, all of these chemicals are electrolytes. So if you look at the very top of the slide where it says calcium, notice how I wrote calcium. It's calcium plus plus. calcium plus plus, and then down here with the sodium, it's Na plus. So these carry an electrical charge. So if we have a muscle that's resting, that's chilling, and then we flood it with these positive charges, what's going to happen is we're going to generate an electrical impulse or electrical activity. And that's what's going to happen with the muscle. So calcium, sodium, calcium. We saw calcium entering the nerve. We saw sodium now entering the muscle. And once again, this is a whole cascade effect, a huge domino effect. You can't have one without the other. So before we move on to the next slide, let's recap all this and do it in order to make sure that you know the proper steps. So number one, the electrical impulse has to come from the brain, from somewhere from the nervous system, and excite those calcium channels in the axon terminal. Once you excite or shock those protein channels, they open up. Once the protein channels that are sensitive to calcium open up, calcium rushes into the cell. Once calcium rushes into the cell, they force those vesicles to move towards the cleft, towards the edge. Once the vesicles get to the cleft or to the edge, they spit out or they exocytose the neurotransmitter. And once again, the neurotransmitter is called acetylcholine. Once acetylcholine is floating around in that synaptic cleft, they just automatically start diffusing high concentration to a low concentration. They diffuse towards the muscle, the acetylcholine receptors on the muscle. And then, once they bind to the receptors open and they let sodium into the muscle cell. So if we're missing any one of those steps, this whole thing falls apart, nothing's going to happen. So if we don't have electricity from the brain to the nerve, the muscle's paralyzed, it's never going to work. If we don't have the calcium outside of the cell, if we don't have enough calcium in our body, this is going to fall apart, it's not going to work. If we're not making enough acetylcholine, the acetylcholine is never going to cause the sodium to rush into the muscle. It's all going to fall apart, nothing's going to work. So we need all these steps in the proper order for your muscles to be able to contract. So now what I'm going to do on the next slide is we're going to talk about this sodium and this muscle excitement. So we're flooding the muscle cell with sodium, and that is going to lead to steps that we've actually already covered. You actually know what's going to happen. Even if you don't know, you know it. But let's go on to the next picture and I'll show you. Okay, so now that we have covered the neuromuscular junction, this is a picture of the muscle zoomed in. And I know it looks like a train wreck right now. Just hang with me and I'll explain what we're looking at here. As you can see, I made another legend or another key to show you what we're talking about. So the big whole thing, what we're looking at under highlighted in red, this is a muscle. All right, we're looking at, actually more specifically, we're looking at the myofibrils at this point. The individual muscle cells and the fibers inside of the muscle cells called the myofibrils. In black, we have these the T tubules, the kind of highways, and you'll see there's one T tubule right here, and there's another T tubule right there, kind of going down the muscle cell. In green, attached to the T tubules, we have those terminal cisternae that make up the triad, and then connected to the triad, it kind of just weaves all throughout the myofibril. That's called the sarcoplasmic reticulum. And then remember, we're going to have our ions. Sodium is what's exciting the muscle cell. So we had calcium, sodium, calcium. We saw calcium get started with the nerve cell. And then sodium is coming in thanks to that acetylcholine. And then we're going to have calcium be released. So what happens is this. Once the acetylcholine activates those acetylcholine receptors, sodium rushes down the cell. Now, sodium rushes down those highways, those interstates, those T tubules. And once again, sodium has a positive charge to it. So this is in the form of electricity. And once again, the purpose of the T tubules is to connect it throughout the entire muscle area, the entire myofibril, so everything is contracting together. So as that electricity goes down the T tubules, what it's going to do is it's going to spread throughout the entire sarcoplasmic reticulum. And I'm not going to draw it on every single one, but basically yellow electricity is going all over the place. And then you know the next step. What is stored inside the sarcoplasmic reticulum? What important ion? So we had calcium, sodium, and this is our last one, calcium. So calcium is stored inside the sarcoplasmic reticulum, and it's kind of trapped in there, if you will. We actually use active transport to pump it back in to keep it trapped later down the road when we relax the muscle. So same thing, same principle. Whenever you shock something with electricity, generally it's either going to move or it's going to open. And when you shock the sarcoplasmic reticulum with electricity, what you're going to get is you're going to get a whole bunch of calcium ions that I did in dark blue. So I'm just going to do this for a second. Calcium ions are flooding the entire sarcoplasm. All the cytoplasm inside of the muscle cell, it's just floating around everywhere. Calcium, 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 calcium. Blue dots everywhere. All right, so why is that important? Now we've introduced calcium, sodium, calcium. Now we can go on to the final slide of this whole drawing series and actually finally move our finger. I know it's been like half an hour since I said we're going to move our finger, but all these steps we need to cover in order for this to happen. This last calcium that's released from the sarcoplasmic reticulum is going to be our key to our keyhole, our keypad, and that's going to help us open up the myosin actin, and it's going to help move the tropomyosin out of the way and get myosin actin together. So let's move on to the last slide and finish up this drawing series. Okay, so then we get to the cross-bridge cycle, and once again, the cross-bridge cycle, or cross-bridge, is where the myosin actin can actually bind together. So you can see in the legend that I drew, we have myosin in red, and remember it's the thick protein with those kind of golf club head looking structures. We have actin in dark blue, and I just drew them as kind of semicircles, being blocked by the green rope-like structure. And remember that the green rope-like structure is called tropomyosin, so I just abbreviated it as TM up at the top of the page. In orange, we have our keyhole, or our keypad, that's called troponin. And then we have our calcium that was just released from our sarcoplasmic reticulum, thanks to the sodium going down those T tubules. So now that we have calcium all throughout the sarcoplasm floating around, this calcium can bind to the troponin, and the troponin, the key to the keyhole. So the troponin is what's going to help us temporarily move the tropomyosin out of the way. And my eraser is not working, but just picture with me that the green rope would be temporarily rotated so the myosin and actin can actually get together. Now, all of this falls apart without ATP. We need ATP in the muscle in order for this to work. So you can have all the calcium, sodium, calcium in the world, you can have all the acetylcholine in the world, but as soon as you're low on ATP or out of ATP, this cross bridge does not work. And that's when exercise fatigue comes into play. But that is how you move your finger one time. That is how you blink an eyelid once. It all starts in the brain, and we got to do all those processes from the nerve to the muscle and then inside the muscle for this to happen. So on the next few slides, we'll recap this with some different pictures and just kind of bring it all together. But the reason why I did this is you can read in the book and you can look at the drawings and the illustrations and yeah, cool, and think that you know it. But reading it and seeing it is very different, especially when you're studying for an exam. So by drawing one thing at a time, with the history of teaching students for several years now, this is what helps stick more. You can look at the pictures in your book, great, but by seeing it and reviewing it like this tends to help you understand it better. So as you review it, I would encourage you to rewind the video, look back, but just make my voice go faster, right? Put it into times 1.5 or times 2 so it doesn't take as long, but you can review it and see these processes go faster and see how this all connects with one another. All right, so let's recap. All right, so reviewing what we just talked about, step 1 in this whole process with the neuromuscular junction and then eventually leading to the cross bridge where myosin and actin get together is excitation. We have to excite the muscle fiber. And you can see in that first bullet point in bold, muscle fiber is stimulated by a neural impulse from a motor neuron. And yes, while that's true, we know that it's not actually 100% solidified truth, right? We are sending an electrical message through the nerve, and then we're releasing the chemical, that acetylcholine, and then that acetylcholine is what's causing the muscle fiber to be stimulated with its own electrical impulse. So it's not a lie, it's not false, it's not wrong, it's just we're being more specific because we are anatomy and physiology rock stars, right? We gotta know our stuff. So you can see, just in review, all this stuff we've covered. First bullet, nerve impulse or neural impulse. It's just the electricity going down the nerve. Goes along the plasma membrane of the neuron. And what that's going to do is open up all those calcium channels we talked about. Calcium's going to rush in. And then calcium is going to cause those vesicles to release our neurotransmitter, ACH. ACH is going to be released, go through the synaptic cleft, and bind to the receptors on the muscle, the acetylcholine receptors on the edge of the motor end plate or the sarcolemma. And then what that's going to happen is sodium is going to rush in. So calcium is here, and then sodium is in the muscle. So calcium in the nerve, sodium in the muscle, and then that sodium is going to create that new electrical charge, which is going to rush down the T tubules and eventually excite the sarcoplasmic reticulum, which is going to release our third ion, calcium. So you can see where we're getting this calcium, sodium, calcium from. And you can look at the picture at the right and you can see all of those steps. You can see all the vesicles and the exocytosis and the acetylcholine receptors and the sodium. Or you can look at my amazing drawings, right? Mine are way better. Not quite. Okay, so then we have contraction, right? So once we release all that calcium from the sarcoplasmic reticulum, we actually have the ability for myosin and actin to get together, because the key is now able to bind to the keyhole. As you can see in bold, calcium is able to bind to troponin. And what troponin does is it temporarily pulls on tropomyosin. It kind of just rotates it about 90 degrees and just gets it out of the way for just a small amount of time, so that myosin and actin can do their thing. Now, once again, this only happens with the help of ATP. So we get the byproducts. We get the building blocks of ATP, if you will, the substrates, which is ADP, which is adenosine diphosphate, which is just adenosine and then 2 phosphate groups. And then we add the third one to make ATP. But with the help of those two, they kind of release the myosin head to bend and cause this sarcomere contraction. So we need ATP and then we break it apart in order for this movement to happen. And remember, anytime you break ATP, this is our energy molecule. So when you break bonds, energy is released. And then we'll have a new ATP molecule that binds to myosin, and it causes to go back kind of in this what they call the recocking position, to where it can do it again and again and again. So we can constantly contract our muscles. For example, if we're walking, your muscles are constantly contracting, relaxing, just every single step you take, right? So ATP is constantly moving back and forth. And you can continue to do this, as I mentioned, as long as you have enough calcium in that sarcoplasmic reticulum being released, and as long as you have ATP. ATP being the main denominator. That's when you can't do any more in a workout. It's usually due to low ATP. Give yourself a minute, 2-minute break, 5-minute break, and you should be able to do more in your workout as your body replenishes the ATP. Now that we have contracted the muscle, we need to relax the muscle, right, in order to avoid fatigue or spasm or anything like that. So several things happen during relaxation. The acetylcholine starts getting broken down and we recycle it. We start moving ions back to their original position. And the main thing that's going to help start the relaxation of the muscle is the calcium gets pumped or actively put back into the sarcoplasmic reticulum. So this is an active transport. We do need ATP to do this as well. But what we're doing is we're stripping the calcium from the troponin. So we're unlocking or removing the key from the keyhole, and then we're throwing the key back in the lockbox, if you will. So taking the calcium, putting him back in the sarcoplasmic reticulum where the muscle was originally resting. Once you remove the key from the keyhole, that rope goes back into place. The tropomyosin is now no longer being influenced by the troponin, and then the tropomyosin starts blocking myosin and actin from getting together again. It blocks that actin open site. So if actin's being blocked, then myosin can no longer bind with actin, and the whole thing shuts off. And what happens is the sarcomere starts extending, going back to its resting state. So, but we're basically just kind of working backwards. If we're removing the sodium, and if you remove if you remove the calcium, is what we're removing. If you remove the calcium, that rope gets put back in its place, and if the rope is back in the place, myosin and actin cannot get together. So that is how we relax. And then if we want to contract, we'll just start it all over again, starting with the brain. We'll send an electrical impulse down the nerve, do the acetylcholine and the calcium and sodium calcium and all that stuff we discussed. So really, really good slide. I would encourage you to take a look at this slide, spend some time on it, study it. This is just showing you all of the things that we drew and talked about in one nice condensed illustration. So the picture on the left, I'll draw a nice barrier. The picture on the left is talking about contraction of the muscle. As you can see down here at the bottom, we're shortening the muscle, contracting it. And the picture on the right is showing how it relaxes. So we're pumping the calcium back into the SR. And the myosin actin here are unattached, so the muscle starts stretching and relaxing. So really good picture. I'd encourage you to spend some time looking at it. Now that we know how a muscle contracts with every single step from A all the way to Z, the remainder of the chapter is just covering general principles of muscle contraction, different types of muscle contraction, and then eventually we'll get into the cardiac and smooth muscle and the similarities and differences between them and skeletal muscle. So the first thing I want to introduce is something called the all or none principle. And you're going to see this again in the nervous system, which will mean something a little different. But for right now, let's talk about the all or none principle regarding muscle tissue. And what it is basically all or nothing. So if a motor neuron, if a nerve cell stimulates the muscle cell or the muscle fiber, that fiber will contract 100%. Pedal to the metal, it is going, the Z discs will move closer together, that sarcomere will shorten, and the entire cell. It's going to happen. And it's going to max out too. It's going to go as hard as it can, which is really interesting to think about. Now, in order for this to happen though, there has to be a certain level of stimuli. So remember, our order is calcium, sodium, calcium. So in order for that first original calcium to be released into the nerve, we need an electrical impulse hitting that neuron. And in order for that neuron to be stimulated, we need a strong enough message from the brain. And what that strong enough message is called is the threshold stimulus. So the nerves need a certain amount of stimuli in order for our body to be aware of it, in order for any muscle activity to be carried out. And a good example of this is, if from a sensory standpoint, if you have an ant crawling on you and you don't recognize it, you don't feel it, and it bites you, right? A red ant. So the ant is on your body. You can't deny that. If you have a bug bite on your leg, something was on your leg at some point in time and it bit you. Right? The reason why you didn't feel it right away, or you didn't see the ant to avoid the bite, is because the ant didn't stimulate your skin nerves strongly enough in order for your brain to be aware of it. So it didn't hit this, what we call this threshold stimulus. If it did hit threshold stimulus, for example, if there was a really large bug, like a huge spider on you, you'd probably feel that, and then you'd be able to react to it. But something as really, really small as an ant, as light as an ant, you don't always feel that, and therefore your nerves aren't excited. And if your nerves aren't excited, your brain doesn't know what's going on until it starts itching like crazy, right? So same thing with the muscle. If the muscle isn't stimulated because the nerve isn't stimulated, so even if your brain's trying to move your leg, if the message isn't strong enough, it's not going to get there, right? I can look at my left foot and say, move, left foot, move. And it's not going to do anything unless I actually send the electrical impulse and force my body to move my left foot. So the message has to be strong enough, and what we call that is a threshold. Now, going back to this all or none principle, it says when the muscle fibers contract, they will contract completely, 100% all force, all or nothing. There's no half contraction. But remember, there are thousands of muscle cells in one muscle. So if I'm picking up a pencil, yes, any muscle cells that I use are going to go pedal to the metal 100%, and they're going to contract as strongly as they can. But how many muscle cells does it really take to pick up a pencil? I don't know the answer, but probably not a lot, right? Let's say 5. I don't know. A pencil doesn't weigh much, and my fingers and my arm, my biceps can move it around pretty easily. So even though I'm only stimulating 5 muscle cells, those 5 muscle cells are working as hard as they can. But if I pick up something like a chair, that's going to be a lot different than a pencil, right? I'm going to have to contract more muscle cells, let's say 50 muscle cells, to contract or to contract enough of the muscle itself to move the chair. So same thing, those 50 are going to be all or nothing. They're either going to move or not move, but the 50 that are moving are doing it to the best of their ability. So that's the all or none principle. So in order for that all or none principle to be effective, we need to have energy. And as we've talked about, energy has to be in the form of ATP for our body's processes. So what we do is we have the adenosine triphosphate and we break it apart into adenosine diphosphate and then a phosphate group floating around. So once you break that bond and create ADP, it releases energy, or the ability to have energy in the muscle cell. However, muscle cells can only store a very small amount of energy in ATP. You can see in your notes here 2 to 4 seconds worth. That's arguable. Some textbooks say it's a lot longer. It can be up to 6 to 8 seconds, and also just depending on what pathway we're talking about. But it's not that long. So what you need to do is you need to be able to replenish ATP very quickly, and we do that thanks to our ATP powerhouse, or our energy factory, the mitochondria. So you can see in the diagrams on the right different ways we make ATP. This is what I was referring to, that this process here called the creatine phosphate pathway, this can help us possibly get more than 2 to 4 seconds, but not much longer. And what we do is we just kind of substitute, we use a creatine phosphate molecule to actually make ATP, and then creatine is the byproduct, the waste product. And then in the second and third photos, you can see other ways that we make ATP. Mainly glucose is going to be our easiest way of making it, and our most efficient way, and our most preferred way with our red blood cells in our brain. So glucose, and then if we have a lot of glucose that's stored, we can store it as glycogen in the muscle and liver. But main thing we're trying to get at is ATP, ATP, ATP. You need to have it in order to make energy. And then this bottom picture, you can see that there are other forms. We can have fatty acids. When you break down glucose in the cytoplasm, you get this thing called pyruvic acid. And all of that actually, or pyruvates actually, and all of that actually ends up in the mitochondria, and the mitochondria, as you can see, can spit out a lot more ATP. So the creatine phosphate pathway is awesome. It's really good. You get 1 ATP molecule per creatine molecule. In glycolysis, you can generally get 2 ATP, mainly specifically talking with no oxygen. We'll talk about that on the next slide when we're running low on oxygen. But as long as we have oxygen, we can continue the process and send this to the mitochondria. And the mitochondria is what can make a lot of ATP, anywhere from 26 ATP to 32 ATP, just depending on what book you read. I'll put 30 to be a little more conservative. But if you send glucose and oxygen through the mitochondria, we get a lot more ATP. And we'll talk more about these specific processes next semester in Anatomy and Physiology 2. But for right now, ATP is absolutely essential for all muscle contraction. Okay, so this is always a fun slide to talk about. Weird some of you out. But another type of contraction, even after death, is called rigor mortis. And this is referring to when the body gets all stiff shortly after death. Usually it takes about 3 to 4 hours or so for this to set in. And you can see what's going on in your notes here, but the main cause, what's actually happening with this, is those pumps, those active transport pumps that actively literally pump the calcium back into the sarcoplasmic reticulum, basically fail when you're dead. So, because we don't have any ATP to fuel that active transport. So what happens is calcium stays in the sarcoplasm and continues to bind to troponin. So yes, even though you're dead, even though you don't have a pulse or you don't have brain waves, calcium is still moving around. Calcium's not alive or dead, right? It's an element, it's atom. It doesn't have metabolic processes. So calcium stays bound to the troponin, which means myosin and actin can still get together. And what happens is we need an ATP molecule to bind to myosin to remove it from actin, as you can see in this first bullet point. So ATP is needed to move it to the resting state. The fact that we're dead, that means we don't have ATP, or we're not making ATP, right? The cells are dead too. So what happens is the muscle fibers are unable to relax and they get stuck in this kind of contracted state, and the body gets really stiff. And sometimes in severe situations, you can actually get movements. You can get jerks, you can get kicks or punches against a coffin. In some really weird situations in the morgue, you can actually get groans and yawns and those kind of things. And it's not that the body's haunted by any means. This is just rigor mortis. Your vocal cords are muscles. So if there's myosin actin still contracting or still connected in the vocal cords, as it starts to relax, you can have some movements and sounds, is all. But the good news is this spasm doesn't stay like this forever. After 2 to 3 days, rigor mortis does go away, and the entire body begins to go limp, and everything just kind of starts breaking down, disintegrating. So, but just a cool type of contraction that we don't always get to talk about. Okay, so how do we actually make ATP? Why do we call the mitochondria the powerhouse of the cell? There's 2 big ingredients that go into this. Number 1, sugar. We need glucose. Glucose is we don't need it, but it is the preferred fuel source. And number 2 is oxygen. When you combine those 2 and you send them to the mitochondria, we can make a lot of ATP. And you can see in the notes right here, necessary for aerobic respiration. Aerobic meaning that we have oxygen available. Shortly we're going to talk about anaerobic respiration, which means there is no oxygen readily available. So oxygen serves as the final electron acceptor, and this will make more sense next semester when we actually talk about the different processes inside of the mitochondria. But basically what we're doing with oxygen and with this whole process in the mitochondria is we're saving our investment, we're saving our money for a later date, and hopefully we'll have more money. So take the hypothetical situation. If I give myself $100 and I give you $100, say that my $100, I go spend it on groceries right away, right? So I buy $100 worth of food. But with your $100, you put it in the bank or a bond or a stock or an investment of some sort, and you let it sit there for 3, 4, 5 years. Hopefully, if the investment did good, after those 3, 4, 5 years, you pull your money out and you have $100 plus the interest that it earned. So maybe you have $120 where I only had 100 or something like that, right? So you're getting more bang for your buck by saving it for a later date. And that's kind of the principle, or what we're doing with oxygen. Is oxygen's kind of the last part of this process to actually help make ATP in the mitochondria. And we do that by creating a concentration gradient, both electrically and chemically. And I'm not going to say any more than that because we're really just not going to get into the nitty-gritty until next semester. So stay tuned. Another important fact with oxygen is that it can be stored in the muscles thanks to a protein called myoglobin. Now, you may have heard of hemoglobin before. Hemoglobin is the protein that binds to oxygen in our red blood cells. So it actually helps the red blood cells carry oxygen all throughout the body. And myoglobin is very, very similar to that. But instead of storing it or binding to oxygen in the red blood cells, it actually binds to it in the muscles. So as the mitochondria is running low on oxygen, we can actually use some of the oxygen in our myoglobin to keep going. So when you start getting tired from your exercise and you start huffing and puffing, you can huff and puff for a little bit longer because you do have some oxygen available to you inside the muscle cells themselves, bound to this protein called myoglobin. And during oxygen, you can see there are different fibers. We're going to be a little bit more specific in just a moment. Instead of saying red and white fibers or red and white meat, we're going to actually put names to them. But basically, you have dark meat and light meat, right? If you eat a chicken or a turkey or, those kind of animals, and those have different characteristics to them. Typically speaking, the red fibers have a lot of myoglobin to them. So therefore, they have pretty good blood flow, a lot of oxygen, so they appear dark when you kill the animal, and even when you cook it, it's dark meat. And generally speaking, this type of fiber has more of an endurance capability. They can help go for long distance. And then you have the light meat, also known as the white meat, and this is more of like the kind of burstful, exploding type of muscle fiber. So you can see in a chicken, for example, less active in the breast. And if you've ever chased a chicken before in a cage or something, they can't fly, right? They have wings, but they can only fly for X amount of seconds, right? But they can run around and try to dodge you from running for minutes, hours even. So they have a lot of endurance in their thighs, their drumsticks, that kind of area. But in their chest muscle and their breast muscle that actually helps flap the wings, they can only go so far for so long, right? So we'll get into more detail on that in just a little bit. So on the previous slide, we talked about aerobic respiration, and the two main ingredients are sugar and oxygen. And with oxygen, you can go for a very long time. You can make a lot of ATP. And you see some of these endurance athletes that can run marathons, that can run ultra marathons, that can do these Ironman, triathlon type things. And it's because they are in this aerobic mode where their body can constantly regenerate ATP to keep them going. However, when you take the oxygen out of the equation, you have what we call anaerobic respiration. And anaerobic respiration means that there is not enough oxygen available. You are pushing your body so hard, so fast, that you can't get enough oxygen into the cells quick enough. So what happens is the muscles fatigue very quickly. And this is what we were talking about with the white fibers, or the light meat. They don't have a lot of myoglobin, so they don't have a lot of oxygen, and therefore they don't get oxygen easily. So with this type of exercise, you can become tired very, very quickly and extremely sore. You can go run 5 miles at a real nice slow jog, slow pace, get a good exercise, good workout in, or you can just run up and down the football field 5 times as fast as you can and be done in about a minute, and you will see greatly different effects on your body and your body's ability to heal, your soreness levels, and your cardiovascular strength. So very, very different type of exercise here. Now, for the longest time, even just within the last decade or two, this term lactic acid was always thought to be negative. This is what causes soreness. You have to minimize lactic acid. Lactic acid is bad. And what we're finding out with recent research is that's not true at all. Lactic acid is just a byproduct of this anaerobic respiration. When you don't have enough oxygen, there are a couple chemical reactions that occur, and lactic acid does build up in the body. But guess what? As you can read in the bullet point, lactic acid is actually used in the body to be converted back to glucose. So this is actually a fuel that the body uses. It's not as efficient as just having glucose right away. So with that research, we're showing, or scientists are showing now, that lactic acid does not cause soreness. Also, lactic acid is a fuel source. So there are some endurance athletes and even sprint athletes that actually take lactic acid as a supplement, because they know that their body can convert it to glucose, which will give them more energy, which will help their performance. So, a big exercise myth debunked within the last few years. Now, with that said, what causes soreness? Well, if you're pushing your muscles to the extreme, if you look at the picture on the right, we're looking at more than likely this is either a 200 meter or a 400 meter sprint. It looks more like a 200 meter based on where they're at. This is a pretty much 90-95% full blown out effort. They are working as hard as they possibly can, pushing their bodies to the extreme limits to get to that finish line. And what you're creating is a lot of tearing of muscle tissue, a lot of myofibrils, a lot of everything's being torn from the microscopic level. And when you tear muscles, just like anything else in your body, you tear your skin, you tear your tendons, ligaments, or cartilage or whatever, pain happens. So that painful burning, cramping, that awful soreness sensation, a lot of times it just comes from you actually tearing your myofibrils, your muscle tissue. The good news is, with the help of growth hormone and just really good nutrition and rest, those muscles heal, and they actually heal stronger than before, as long as you didn't completely tear them. So one more thing that I want to point out with anaerobic respiration is, just like aerobic respiration, I mean, there's going to be an oxygen debt. An oxygen debt means that your body used a lot of oxygen during exercise and it needs to catch up. But it's pretty severe and very, very noticeable in anaerobic respiration. So these athletes right here, once again, let's say it's a 200-meter race. They can run this anywhere from 20 seconds to 22 seconds. Unless you're Usain Bolt, they don't really run it under 20 seconds. So for 20 seconds, they're pushing their body to the absolute max, and that's it. And then they're done, 20 seconds. But if you watch them after the race, and if you look at the interview 3 minutes after they've completed the race, even the guy who finished in last place is still huffing and puffing. So they worked out for 20 seconds, and then 3, 4, 5 minutes later, their body's still trying to catch up on oxygen. And we see this with exercise all the time, right? But it's just very, very noticeable with anaerobic, because their bodies weren't able to get enough oxygen while they were pushing themselves. So that's anaerobic respiration. The mitochondria isn't really involved at all here. We're getting very limited ATP, and that's why these guys can only go so long. They can only go for 20 seconds. Anything further than this race, you're not using anaerobic respiration the majority of the race. You have to use a different type of energy source, mainly aerobic. So, all right, moving on to contraction principles. Let's talk about different forms of contraction of muscles. Let's first introduce what a motor unit is, and you've actually somewhat been exposed to this already with the neuromuscular junction example. A motor unit, as you can see, is simply a motor neuron, and a motor neuron is just a neuron that talks to a muscle, so it causes a motor movement, it causes movement of something. So it's a neuron that talks to muscle cells or muscle fibers. And as you can see in the parentheses, it ranges from a few dozen to several thousand in the body, depending on the size of the muscle and the motor unit itself. So if we're talking about really, really small muscles, we're going to have few fibers, and we call that a small motor unit. And the smaller the motor unit, the more precise, the more accurate the movement's going to be. So picture your finger muscles, that you can move your fingers very, very quickly and very precisely, right? You can type a text message with your eyes closed, or you can type on the keyboard while looking at the screen instead of the keyboard, right? So your muscles are trained to move in very precise movements very quickly. Would be an example of a small motor unit. And then if you have several thousand cells being affected by a motor neuron, that would be just simply a large motor unit. And that's going to be your really, really bulky muscles, such as your quadriceps muscles. Now you think about all the cool things your quadriceps can do, but it's really not that much. Your quadriceps really just extend your knee, and that's it. So yes, you can do neat things such as running and jumping and climbing with your quadriceps, but really what's helping you land and move in all those different directions would be your small, intricate foot and ankle muscles. So your quads would be considered a very large motor unit that helps a large muscle do its main job. So a motor unit is a nerve talking to X amount of muscle cells. Okay, so now we're going to introduce a few different types of contractions. And the first one's pretty basic. It's called a tonic contraction, or some people say tonic, same thing, it's okay. A tonic contraction, as you can see, deals with your muscle tone. And this is not exactly what you think of when you think of muscle contraction, but if you reach down to your calf muscle and just grab your calf muscle and just kind of, you can massage it a little bit, but just kind of feel what the muscle feels like. And even though it's relaxed, like you're not running or walking right now or plantar flexing, you can feel that it's still got a tone to it, right? It's still kind of, it's not exactly mushy. There's muscle and it's kind of tough in there. And you can do this with all your muscles. You can feel your shoulder muscles. Even if your arm is just hanging by your side, your shoulder muscles are still somewhat engaged at all times. And this is what we call a tonic contraction. So all of our muscles in our body, for the most part, have this always kind of partial contraction to them. So they're not completely ever limp. And there's several reasons why. Number one is only a few of the total, this SMF right here, this is skeletal muscle fiber. It's just abbreviated, so skeletal muscle cell, contract at the same time. Remember, we talked about the all or none principle, right? That when a muscle cell contracts, it's going to contract as hard as it can, 100% pedal to the metal. So if we have 10,000 cells in a muscle and only 3 are contracting pedal to the metal, the other 99,000 plus are resting. So as soon as those 3 are done contracting pedal to the metal, well, guess what? They get a break. And then the body, the nervous system, will choose another motor unit, another 3 or 4 muscle cells to contract. So at all times, your muscles have a little bit of tone to them. And this is mediated by something called a spinal reflex. So this is a nervous system control. And as you can see, stretch receptors in your muscles and tendons detect or monitor how much your body is stretching. And then when out of range, the nervous system kind of kicks back and says, hey, don't go that far, or contract. And we get this, just perfect example is go down, reach down and touch your toes. Stand up and then try to bend down and touch your toes. And some of you can touch your toes, and that's awesome. That's great. Some of you can't. Some of you, you'll actually bounce upward, whether it's due to pain or your muscles just won't allow you. For those of you who can touch your toes, try to put your palms flat on the floor. and then you'll kind of get that little kickback. Every single one of us has our own kind of medium to where the nervous system says, hey, that's too far. Your muscles are only supposed to go this long. So what this results in is a tone or a firmness in your muscles at all times. Once again, feel your calves, feel your back muscles as you're sitting in your chair or lying in your bed. Feel the muscles in your neck. They all have somewhat of a tone to them. So a lot of these contractions, especially in your hips, glutes, pelvis, lower back, and abdominal area, the main job for these muscles in your neck as well is to maintain posture. So if you're sitting, standing, or even if you're lying down on your side, some of these core muscles are still engaged to an extent. And you can read at the bottom bullet point, they continue as long as one is awake and conscious. And I would even argue that. I would debate that and say, even when you're sleeping, you have tonic contractions. Now, there is a phase in your sleep where your body is completely paralyzed, completely limp, and you can't move. But even throughout your sleep, there is contraction of your muscles, and you know, everyone. That's why with babies and with kids, we have cribs, right, or we have barriers so they don't fall off the bed. As adults, we get a little better with not moving as much and being aware of our bed situation, but there is still moving and tossing throughout the middle of the night, and those muscles are contracted. A lot of folks sleep in the fetal position. Right, their knees are kind of crunched, and that their upper body kind of leans forward, and they sleep in that kind of curled up position. Well, the only way you can sleep in that curled up position is if your muscles are contracting. So I would kind of argue that last bullet point, but just to give you an idea, your muscles are working all the time. It's just very different than, you know, lifting a box or running up and down the stairs. So this kind of staying still, maintaining posture type of contraction is called a tonic contraction. Now, a specific type of contraction where we are actually moving the joints and the muscle is changing shape, so we are actually lifting a weight or lifting a book or lifting a box or whatever we're lifting, is called an isotonic contraction. And the isotonic contraction, as you can see in the definition, is the tension is the same. So say that we're lifting 20 pounds. When we're lifting 20 pounds and we put it down and we do another rep, it's still 20 pounds. So that doesn't change, but the length of the muscle changes. And a good way to remember this is you're moving the joint. So if I'm flexing or extending my elbow, or if I'm plantar flexing or dorsiflexing my ankle foot region, that is an isotonic contraction. The muscle is changing shape. So I always talk about measuring the biceps brachii muscle, right? Take a tape measure and measure your biceps from where your scapula and your humerus kind of meet, go all the way down to the radius area, and then flex your elbow, make a biceps, kind of, you know, flex your guns. And you'll see that that muscle actually gets shorter. It kind of crunches up, it bunches up. And whenever you see that bunching up action, you're moving the joint, and therefore that is called an isotonic contraction. Now, the second bullet point, it says occurs when little or no load is placed on the muscle. That's not, you can put a lot of load on the muscle. It's just your body's still able to handle it. So for example, if I'm bench pressing, let's say, I can't bench press much, but if I'm bench pressing 160 pounds, I can move that up and down, up and down. That's an isotonic contraction because my joints can move it. But if I'm bench pressing 200 pounds, I can't do that. So I get stuck. So because the load is too much for my muscles, I can't move the joints, and therefore I can't move in an isotonic contraction. So as long as the weight isn't heavy enough, you can move the joints in an isotonic fashion. So the key thing to remember is the muscle changes, the length changes, and therefore your joint does move. You do bend or straighten something. And kind of the opposite of an isotonic contraction is what you see here, an isometric contraction. So what's happening with an isometric is we are not moving the joint, and therefore the length of the muscle stays the same, even though tension may increase. So there are two examples of a situation where we can illustrate an isometric contraction. Number one is holding still. So for example, instead of taking the dumbbell, you can see in this picture, these top two illustrations that I'm circling in red, this is isotonic contraction in red. So what happens is you're moving the dumbbell up, you're flexing your elbow, and then you extend your elbow, and then you flex, and then you extend. So you're moving the elbow. That's the previous slide. Isometric is this bottom illustration. You can see that the person is just holding the weight and not moving. Now, if you grab a weight, I don't care if it's 5 pounds, 20 pounds, or 100 pounds, and you hold it in that position, your muscle is still working. I promise you, your muscle will get tired, and it will be sore possibly the next day. But you're not moving the joint. So that's an example of an isometric contraction. Anytime where the body is stabilizing or the muscle is stabilizing, you're not moving. So great examples of isometric contraction are planks, wall sits, those kind of things where your body is, you're trying not to move your body, right? The whole purpose of a plank is for core stability, and you're trying not to move your arms and legs and your hips. If you are moving, that means you're not doing it right. Another example of an isometric contraction is when something is too heavy. The load is too much and you get stuck pretty much. So the example that I used in the previous slide was the bench press, right? If I put more weight on the bench press than I can handle, I'm going to do an isometric contraction. So that I'm going to lift the thing off and it's going to get stuck on my chest. And no matter how hard I push, I'm not going to be able to get it off, right? So therefore, my joints aren't moving. Even though my muscles are moving, I'm trying to move the weight, but I'm stuck. That's another example of an isometric contraction. Something is too heavy and you just can't move it, right? So trying to lift a car. You're putting a lot of strain on your muscles. You can get a great workout trying to lift a car, but if you're not lifting that car, your joints aren't moving and neither is the car, right? So what's happening is that actin myosin, even though the myosin is grabbing onto the actin, it's not moving. It's not getting swung towards that midline. Now, so when we're talking about personal training and exercise, and well, which one's better for me? Do I want to do aerobic exercise or anaerobic exercise? Do I want to do isotonic contractions or isometric? And the answer is, unless you're, well, even if you're training for something very, very specific, you want to be good at both. You want to have aerobic capacity and anaerobic. Aerobic is going to help you with cardiovascular fitness and endurance, and anaerobic is going to help you with cardiovascular fitness and strength and power. Isotonic contraction is going to help those tendons and ligaments and those joints be healthy and help you move. Isometric is going to help with a lot of stability. So you want both in your workout. And as you can see, the last bullet point, most body movements are a combination, right? So even as I'm walking, yes, I am flexing and extending my knee. My quadriceps and my hamstrings are moving with every step. My calf muscles and my shin muscles are moving, contracting isotonically with every step. But what's keeping me balanced are my glute muscles, my hip muscles, my abdominal, my lower back muscles are keeping me upright while I'm walking. So your leg muscles are isotonically contraction and your core muscles are isometrically somewhat contracting. So you do this even if you don't realize you're doing this all the time. Okay, so we have a few more types of contraction that we need to introduce. Number one is a twitch contraction, and you can see it's a pretty simplistic definition. It's a quick jerk of a muscle that occurs when an entire muscle is excited at the same time. So key word here is jerk, quick jerk, meaning it's singular. So a true definition of a twitch contraction is just one jerk of a muscle. Now, this doesn't really happen unless we're in a laboratory setting. For example, when you're walking, when you're talking, when you're moving, when you're moving your arm, your nervous system has to send multiple messages to those muscles in order for that to work. A true twitch is just one stimulation to where we have one excitement of those ions, those sodium ions rushing into the muscle, and then that's it. So the only way you actually show this or illustrate this is you're in a lab and we have a probe that sends electricity to the muscle, and the probe is controlled by a computer, and you only shock the muscle one time. So we can introduce multiple twitches at a time, but that's more where we call they have different names for those things. So a true twitch is just one contraction due to one stimulation of the entire muscle. Okay, and then another type of contraction is called trep contraction, also known as the staircase phenomenon. And this is, as you can see, it's a step-like increase of strength. So what happens is this is what we call the warm-up type of contraction. So if you take a look at the picture on the right, the amount of tension is on this y-axis right here. So it's how well you can tense or contract that muscle. And then on the x-axis is time. So what we're doing is we're contracting the muscle, about 8 times in this picture. So picture waking up first thing in the morning and trying to make a fist, right? It's kind of hard to do, right? Right when you wake up, all your muscles are kind of weak, right? So the first time you make a fist, your muscles can only contract so much, and then when you relax them, they just go back down the rest. And then you try to make a fist again, and it's still kind of hard, but it's probably a little better than the first time. And then you do it again, and it gets a little better, and you get where I'm going. With every contraction that you get, the muscles get a little stronger, and then here you can make a full fist that's strong just like normal throughout the day, right? So what happens is we are resting the muscle, but we're repeatedly stimulating it to kind of wake it up and get it at its full strength. So the whole principle behind it is the more you contract it after a few times, the more forceful this muscle can contract. So basically you're warming it up. And this is the whole application behind why athletes do a pregame. Why do they start jogging and moving and shooting hoops or whatever sport they're playing? Why do they warm up before the event? Well, one, it's to get the blood flow going so they don't pull anything, right? It's to get their bodies somewhat warm and ready for action. But from a muscular standpoint, it's this. So say that we are playing basketball. If I need to jump 30 inches in the air to dunk a basketball, it's kind of hard to do that unless you're a freak athlete. It's kind of hard to do that right when you just stand up and get off the bench. But if you start real light and just do a layup or just do some jumping exercises and you do that 15, 20 times, then yeah, you have no problem dunking in the game. So you got to warm up the muscle, and as you warm up the muscle contracts more efficiently. It contracts better. Now this will continue to happen. This trep, this staircase will continue to get better and better, and the muscle will continue to contract more optimally until you get tired. There's a limit to everybody, right? So you will exhibit fatigue eventually. You can only warm up for so long. You can only play basketball or tennis or whatever we're playing for so long. And eventually this will decrease, right? We're not robots. But certain people can go and go and go and go for a very long time. Eventually everybody will tire out though. So that's trep. And one more type of contraction is called tetanic contraction, also just known as tetanus or tetany. And this is a contraction where you're not giving the muscle enough time to relax. So it's a prolonged contraction, and you're continually shocking it with that electricity. You're constantly sending those nerve impulses to the muscle very, very rapidly to the point where we do not have time to rest. So if you remember with the trep example on the previous slide, the way the graph kind of looked was like this. The muscle went up and then it came back down the rest. And then it went up a little higher and it came back down the rest, and then a little higher and rest. If you notice the pictures on the right, we're not going back down the rest here. We're actually building on it. So we're sending multiple waves of electricity to the muscle, and that's why this form of contraction is also known as multiple wave summation. Summation meaning summing, like we're adding these waves on top of one another. So if you look at the first picture, this is actually what we called unfused tetanus or incomplete tetanus, but you contract the muscle, and before it has a complete chance to relax, we are contracting it again and again and again. And you're kind of seeing the same effect as TREP. You're seeing this staircase phenomenon, but this is not really dealing with warming up. This is dealing with you're not giving the body enough time to relax. So this is an example, kind of like running, if you will. You just keep going and going and going and going. Your muscles are constantly working. So it doesn't exactly create a series of, it does. It creates a series of twitches together, but remember the true definition of a twitch is just a single contraction. And this can be so extreme. If you look at the picture on the far right, this is called fused tetanus or just full tetany. And where there is no building, it's just straight max effort. So the muscle has zero time to rest. And this is like, for example, if we're moving a couch and you're holding the couch up and we're going down the stairs, you don't have a choice. You have to hold on to that couch. If your muscles let go, we're both falling down. We're going to hurt our back and then we're going to fall down the stairs, right? So your muscles are tensed and they are not getting a break at all, pretty much. So big keywords here. It's also known as multiple wave summation, and we are not giving the muscle enough time to relax before the next contraction. So we're really taxing the muscle. Now that we've covered different types of contraction, we can talk about a couple more principles, relationship rules that kind of oversee the different types of contraction our body can do. So one here is called the graded strength principle. And the big thing with the graded strength principle is that your muscles can contract with different levels of strength at different times depending on your needs or what you have available in the muscle. So let me explain that. What this does is it allows you to match what you need to do with how you have to do it, with the task. So for example, if I am arm wrestling somebody, say my age and my size, I have to put in X amount of effort, right? I probably have to put in 90 to 100% effort in order to beat somebody my size and my age. But if I'm arm wrestling my 5-year-old nephew, I'm going to break his arm if I go full 90%, 100% full force, right? I have to kind of match that. I have to be a lot lighter and more gentle if I'm arm wrestling or doing anything with him, right? So the big thing, what controls this, is how many fibers you're actually contracting, or what we call recruiting. And a lot of this is controlled by your brain, right? My brain is going to tell me, hey, this is a 5-year-old, don't hurt him, or be gentle and let him win, right? Versus somebody my age, I can't, if I put the same amount of strength or force as I would with the 5-year-old, I may have my arm broken because the person arm wrestling against me is going to put so much more force against me than I'm putting against him. So your brain can dictate this. Your brain can change this. And we see this a lot at the gym too, not just lifting weights, but with different stimuli around you. I'm not going to lie. I'll try not to be like prideful or anything, but when I'm lifting weights by myself and I'm lifting weights with my wife right next to me, there's a little difference there, and I try not to be prideful, but like I can't explain it other than I kind of want to show off for my wife. So if I'm doing this exercise at 50 pounds, if my wife is right next to me and she's doing something else or doing the same thing, I'm probably going to do 55 or 60 pounds, and I'll probably be able to do it even though I'm used to 50. With that extra motivation that your wife is watching, you can kind of psych yourself up and get your brain recruiting more muscle fibers. And then another thing is I don't really like loud, crazy music. I'm a very chill guy. I like my Kenny Chesney, my Ed Sheeran, my who else have I been listening to a lot? Morgan Wallen and Florida Georgia Line. Like real chill. I listen to a lot of spiritual Christian music as well. Very, very calm music for the most part. But at the gym, I listen to very, very, I guess you can say angry music. I listen to rock music. I listen to Trapped and Linkin Park and Stained and some of those groups you probably never heard of, but they were big in my time. I listen to music that gets you kind of mad, kind of pumped up. And the reason why is if I'm listening to this nice soft country music, my brain probably isn't going to be recruiting as many muscle fibers as I can versus if I'm actually angry or simulating an angry environment to where I'm actually taking the anger out on the weights or whatever exercise I'm doing. So there is a science to that. There are actually experiments. I'm not just being weird. There are experiments where people listen to classical music and do a workout, and then they listen to, punk rock music and they do a workout, and there's a great difference. So I'm not telling you to listen to angry, music with very provocative words in it or anything like that. I'm just saying the greatest strength principle is dictated by how your brain controls your muscle connections, your synapses. Other things that are involved with this is the amount of ATP. So if I'm arm wrestling somebody my size, I have to have the ATP available, right? If I just did a 55-minute workout and then my friend says, hey, let's arm wrestle, I'm probably not going to be as good as I was about 55 or 45 minutes ago, right? I'm going to be a little more tired. I'm probably going to have a hard time winning. And then, as I mentioned earlier, the load imposed on the muscle. It just depends, right? If I'm lifting 10 pounds, I can lift that with a couple fingers. I don't have to use my entire lower body, my back, all my arm muscles. I can lift that with a couple fingers and, twirl it around a little bit. If I'm lifting a 40-pound dumbbell, I better bend my knees and bend my hips and secure my abdominal and back muscles to make sure I don't have a hernia, right? I have to be careful the heavier you go. So this is showing you that depending on the body's needs, depending on what you're lifting, what you're doing, and how your brain is connecting with your body, the muscles can contract with different strengths. So that's the graded strength principle. Okay, on this slide, nothing really crazy with new information or anything like that. A lot of this is kind of common sense, to be honest. If you just read it, more muscle fibers contracting at the same time yield a stronger muscle contraction. And that makes sense, right? If I'm lifting a pencil, I'm only going to use a couple muscle fibers. If I'm lifting that 40-pound dumbbell, I have to use a lot. And when I use a lot of muscle fibers, I'm going to use a lot more strength, and the muscle's going to be stronger, contract more. So it depends on how many motor units you're recruiting. And remember, a motor unit is a nerve talking to different muscle cells. So the more nerves I recruit, the more cells I recruit, the stronger the contraction is. So the more intense and frequent I send those electrical messages, and then you can just keep on reading, the arrows pointing to this next bullet point, the more motor units I can activate. The more motor units I can activate, the more skeletal muscles contract. And the more skeletal muscle fibers, cells I contract. And the more skeletal muscle cells I contract, the stronger the contraction. Up to a point, right? We're only so strong. I can contract and listen to all the crazy music I want. I'm not going to bench press 250 pounds. It doesn't, even if you give me steroids, or I'm not going to do it. It's not happening, right? So it can only it can only happen to your body's own capability. And also the initial length of the fibers. This will be very important too. We talked about stretching the fibers as well. If the fiber is overstretched, it's not going to work properly. If it's not stretched enough, it's not going to optimally contract. And I'll show you a nice little graph in just a little bit coming up soon. And here's the chart that I mentioned on the previous slide. We've actually already talked about this length-tension relationship previously. I mentioned that in order for the muscle to maximally contract, we have to stretch it, but not stretch it too far. So there's this kind of sweet spot, this middle ground to where you're really going to get the most bang for your buck, or the maximal contraction, when you have this kind of middle ground. So take a look at the chart for me on the right, and let's introduce what we're looking at. Once again, on the y-axis, we're looking at tension. How tense can you make the muscle, or how well can you contract the muscle? And then on the x-axis, we're looking at sarcomere length. So how stretched or not stretched are those z-lines or z-discs? So you can see that when we have decreased length, which means this is not a lot of stretching going on, this is a, let's say, very, very tight, let's say. So the muscles are tight, they're constricted, they're not very flexible. What you can see is we only have so much tension. At this point, we're only meeting 20% tension. As we stretch the muscle, it goes up in tension. And as we continue to stretch, it goes up in tension and up in tension. So then to where we get to this nice sweet spot where we can maximize 100% of the contraction with a certain amount of stretching. So going back to the jumping on the table or jumping on the chair analogy, right? In order to jump up on the table, or whatever analogy I used, you have to bend your knees and bend your hips and even bend your back a little bit to optimally jump on the table. If you just jump on the table and you're just standing straight-legged with no bending, good luck. You're going to get 1 inch off the ground, right? Your muscles are not going to be able to contract well. If you stretch too much, say go into a squat and you just basically go all the way as low as you can to the ground to where your glutes are basically touching the floor, you're not going to be able to jump up on that table. So you're just going to get stuck in that squatted position. So what you need to do is you need to bend your knees, bend your hips, bend your ankles, bend your back, all kind of in this middle ground, and that's going to help you explode up to jump on the table. And this isn't just jumping. This can be any type of analogy. Starting a race, right? The racers have a certain position that they're in. Even swimmers, right? When they jump off the board and jump into the pool, they're in a certain crouched position. They're stretching their muscles to a certain extent, but they're not overstretching them. Because look at what happens when you overstretch the muscle. That myosin actin start getting pulled apart, and you lose strength significantly. Big time. So there is a trade-off here. We want the muscles to have stretch to them, but not too much stretch. So that's called the length-tension relationship. And we're going to introduce this again in next semester in the heart, and the heart's the same thing. We want to stretch the heart muscle so it contracts and pumps out the blood that we need to be pumped throughout the body. But if you overstretch the heart's not going to be able to contract forcefully, and you're going to get fluid backed up into the lungs, and your heart's going to shut down, and we're going to have some serious issues. So skeletal muscle, cardiac muscle, it's all the same regarding the perfect sweet spot of this length of the sarcomeres. Okay, we've talked about a lot of different types of contraction over the last few slides. So this slide is a really good way to just kind of slow things down, take a breath, and review, and make sure that we're understanding the different types of contraction that we talked about. So let's just start with number 1, and you can kind of just work with me. Well, actually, I would encourage you to pause the video and do this on your own. Let's do that. Pause the video, do this on your own, and then come back and compare your answers with me and work together with me. All right, so now that you've hit the play button again, let's start with number 1 and look at the possible answers. So a tonic or tonic contraction is number 1. And remember, with tonic contraction, what we're looking at is your tone. This is basically when you're just hanging out and you feel grab your whatever muscle, your triceps muscle, even though you're not extending your elbow, there's still tautness, there's still some tension in that triceps muscle, even if you're not using it. So look at all these different answer choices, and what you'll arrive at is A doesn't look really that great, B looks a little funny, C's not quite right, D, that looks pretty good. Maintain posture, right, with tone. So we talked about sitting upright or standing upright or walking. These muscles are undergoing something called a tonic contraction. So what we'll do is put D for number 1. That should make sense. Number 2, isotonic contraction. Remember, isotonic is you're moving the joint. You're actually changing the length of the muscle. So A and B look pretty promising. A is something that's too heavy. B is the length is changing. The length of the muscle is changing. So B looks good. Isometric is where you're contracting but the muscles are not changing. And the examples I gave you was a plank, was a wall sit, or lifting something too heavy, right? My bench press where I get stuck, that kind of thing. You're not going to be able to move the joints and the muscles the way you want to. So that would be an isometric contraction. So then we're just down to the last 3. We have twitch, and remember, twitch is just a singular movement. So the only one that's singular, you see C is prolonged, E is gradual. So a quick jerk of a muscle would be a twitch. So we'll put E. And then we just have it narrowed down between trep and tetanic. And remember, trep is the warm-up one. That's kind of the building up one. I mentioned the squeezing your fist in the morning. The more you do it, the stronger it'll eventually get. And then that leaves us with C for tetanic, which is the prolonged muscle contraction. With repeated stimulation, we don't allow the muscle enough time to rest. So hopefully your answers line up with mine. And if they don't, no big deal. Just rewind the video and review these studies a little bit more and then compare your answers down the road. And just make sure you understand this before your next exam. All right, moving on, let's talk about the different types of muscle fibers that help us do certain activities. So previously we talked about dark meat versus light meat. We talked about the red stuff in the chicken and the light stuff in the chicken. We're going to be a little bit more specific now. All right, so with this slide, we've talked about the majority of these terms before, so I won't spend too much time on it. Just going to draw a little barrier between the two. So with the red meat, or what we call the dark meat, we're going to introduce one new word, and that's called oxidative. So we have oxidative fibers, and all that means is they use oxygen very efficiently. So they are therefore aerobic, right? Using aerobic respiration, which means we have a lot of myoglobin. Myoglobin, once again, is that protein found inside of the muscle that binds to oxygen, holds on to oxygen. And because we have a whole lot of oxygen inside of the muscles, we also have a whole lot of mitochondria that can make us a lot of ATP. So these guys and gals, they can go for a very, very long time. They're what we call fatigue resistant. And you can take a look at these runners, and these are definitely long distance runners, just based on their body build, based on their outfits. They're going for a long time. I don't know if they're starting, finishing, or in the middle, but they are, I mean, these are marathon runners, potentially. You can look at their body build alone, their muscle physique, and know that they are long distance or endurance runners. You look at the bottom, and we have the white meat or the light meat, and that's what we call glycolytic type fibers. And we're going to have not that much oxygen, so therefore we call this anaerobic respiration. And because we don't have a lot of oxygen, we don't have a lot of myoglobin, nor do we have mitochondria. So we're not going to be making that much ATP. So although these muscles are very strong and powerful, they get very, very tired very quickly. So you think of, I mean, you look at this bodybuilder guy or powerlifter, whatever he is, pretty jacked dude. You tell that guy to go run a half marathon with the guys on top, he's just going to laugh at you. He's more than likely he's not going to be able to do that. Or if he does do it, he's not going to be anywhere near the guys on the top picture, right? You take a football player, whoever your favorite football player is, and tell them to go run a marathon. They're going to laugh at you because football is designed to be very explosive, powerful. You do pedal to the metal, maximum effort for a couple seconds, and then you take a break, right? The play's dead, and you got to wait for the ball to be set up, huddle up, get the play, and then you do it again. Unlike a sport like soccer or running cross country where it's just constant movement, right? Different body types, different athletes. So on the next slide, we're going to just go into a little bit more detail and talk about the three main types of muscle fibers that exist in human muscles. Okay, these are the different types of fibers. We have a total of 3. And remember, fast and slow twitch just refers to the speed of the contraction, right? If it's a real jerky, quick movement, that's going to be a fast twitch. If it's nice and steady and slow and fatigue resistant, that's going to be a slow twitch. So 3 types, and depending on what book or what article or what journal you read, a lot of times they put numbers to them. They put type 1 and type 2A and type 2B, type 2X. And there's just so much debate and disagreement within the exercise physiology community that I don't even use those numbers. We're just going to not even talk about them because some folks think that certain fibers aren't even found in humans, while other folks say, oh, this is in power lifters and weight guys or big, heavy strong football players. And some guys say, no, this only exists in like small animals. So it's really up for debate, but there is no argument with these 3 names in humans. So in no particular order, the first one I want to introduce is slow oxidative. And these are the ones that are going to use oxygen, so they're going to have a lot of myoglobin, and therefore this is going to be more of the dark meat. This is going to be the red. And once again, the red stuff is slow. Okay, I'm going to put A, B, and C just to not confuse you with the numbers. A, B, and C. So this, once again, less powerful contractions, so they're not as strong, but they can keep going and going and going and going like the Energizer Bunny. So these are the marathon runners, the triathlon guys. I'm just going to put 26.2. That's how many miles are in a marathon. But these are the very long distance folks that are specializing in slow oxidative. Fast oxidative is kind of in between the 2 extremes. So technically it's really called fast oxidative glycolytic. It's FOG, but let's just keep it simple and call it fast oxidative. And this is going to have a little bit of both. It's going to have a little red and a little white. So it's going to, if you combine red and white, you're going to get kind of more pinkish tint to it. And it's going to have characteristics of being fast, but not that fast. So this is just kind of in the middle. And then we have fast glycolytic. And fast glycolytic is going to be the white fibers. And this is going to be the very powerful stuff. This is going to be the very forceful, jerky contractions. Now, before we move on to the next slide, I want to make this very clear. All of you, all of us, we have a combination of all 3 types of these muscles, okay? So if somebody says, I'm really good at slow twitch or I'm really good at fast twitch, yes, that is possible. But you have all 3 of these in almost all of your muscles. So let me explain. Say that we are golfing, right? With golfing, it's a lot of standing around. So you can use all three of these muscles while golfing. First off, you're standing because it's not your turn yet. So you're watching your buddy hit the golf ball. And as you're standing, you're using those tonic contractions. So all those abdominal muscles, those glute, hip muscles, lower back muscles that are helping you stand or sit in the golf cart, either one, those are your slow oxidative fibers. So those are the ones that are going to help you sit and stand for a very long time, right? Golfing takes a while, so you need some endurance. All right, so your buddy hits the ball, and now it's your turn. So you're going to walk up to the tee. You're going to slowly start moving. And as you're moving those muscles, you're using a little bit of fast and a little bit of slow, right? Because you went from a standing still position to let's move. So you are using a couple of those. And then if anybody's ever watched a golf swing, at least from like a driver standpoint, like they're teeing off, it's a very violent, jerky movement. It's not, I mean, you have to be strong to play golf, right? I'm horrible at it, but it's a nice little rock back, and then they explode through the ball, and they hit that thing hundreds of yards. The good ones do. So that would be our fast, powerful, jerky movements. So all of us have all three types. Now, the big difference between myself and Usain Bolt or the marathon runners or Michael Phelps or any, LeBron James is I don't specialize. I have just a big mixture of all three of these everywhere to where I'm not really good at anything. Where if you are an endurance runner, if you're a marathon runner, you have all three types, right? Those marathon guys can sprint towards the end. They book, but they're really good at the long distance stuff. So most of their fibers are slow oxidative, and they train those slow oxidative to be better. You look at a guy like LeBron James, the majority of his body is the fast glycolytic. Well, actually, basketball's that's a bad example. Excuse me. Let's just go with the slide. Power lifters, heavy bodybuilder guys, that would be more fast glycolytic, powerful, explosive. LeBron James has to have a little bit of both, fast oxidative and fast glycolytic. So we all have mixtures. We're all mutts, but the extreme athletes, the folks that you'll see in the Olympics, those are just more extreme. They specialize in one of these three, mainly the two extremes, either slow oxidative or fast glycolytic. All right, almost done. The last part we have to discuss in this chapter is cardiac and smooth muscle. So we've talked all about skeletal muscle up to this point. We're just going to talk about some similarities and differences between these two involuntary types of muscle. And then we'll do a quick review, and we will be done with muscle physiology. All right, so with cardiac muscle, nothing new here. This is all review from the tissue chapter, so I'll fly through it. It's only found in the heart, hence the name cardiac. The job of the heart muscle is to pump blood throughout your entire body, so your body gets oxygen and nutrients. And as I just mentioned, it's involuntary, right? We cannot control your heart rate. Your heart is beating right now, whether you like it or not. And here's a picture of cardiac tissue. And we will talk about the characteristics of the micrograph on the next slide. So a big difference between skeletal and cardiac muscle under the microscope is skeletal just seems a lot more organized. It's a lot more neat. Cardiac is a lot more branching going on. But similar to the skeletal muscle, we do have striations. We do have those stripes, that light, dark, light, dark, light, dark, altering proteins appearance. So because we have that light, dark, we do have Z-discs. And if we have Z-discs, we do have sarcomeres. So the sarcomeres kind of work the exact same way as skeletal muscle. They get pulled closer together, which causes contraction of the cell, or of the sarcomere, excuse me, which will eventually cause contraction of the cell. Now, big difference between skeletal and cardiac is cardiac muscle only has one nucleus per cell. So this is what we call uninucleated. And that's typically of every cell in the body, right? Skeletal muscle is really weird being multinucleated. The T-tubules are the exact same. We need to be able to get that message quickly throughout the heart. So we do have T-tubules. And we also do have a sarcoplasmic reticulum in the heart. However, it's just not as used, it's not used as extensively. It's not as important. Now, as you can read, the sarcoplasmic reticulum, it still does store a little bit of calcium. But most of the calcium regarding the heart is actually brought in from the outside of the cell. So the sarcoplasmic reticulum is there, it's just it doesn't play as big of a role, is what I was trying to say. Another little factor is we don't have the triads, we just have one terminal cisterna on one side of the T-tubule, so instead of triads we call it a dyad. So it's not a sandwich like it is in skeletal muscle. I mentioned the branching is a little different compared to the skeletal muscle. Skeletal muscle is really neat and organized, and the cardiac muscle has all these different branches. If you go back a couple slides, you can really see the picture. You can also just take a look at this representation where they just kind of branch off of each other. Still connected, but they're just not as neat. And what helps these branched cells communicate very efficiently are something called intercalated discs. And what these are tiny little gap junctions. So it's a connection point with this tiny little gap, and that gap allows the ions, those electrolytes, the calcium, sodium, potassium, to move back and forth. So even though they're not as neat as skeletal muscle, they are all still connected, and they can communicate with one another through these tiny gaps called intercalated discs. We also have a lot of flexibility in the heart muscle cell compared to the skeletal muscle cell. We call those desmosomes. If you're looking at this picture, you can see a desmosome just gives a little bit more flexibility so the heart can expand with blood and then forcefully contract. All right, very cool slide talking about the differences between skeletal muscle and cardiac here. Cardiac, very unique. And we'll talk more about this next semester in A&P2 in the cardiac chapter, but two big important concepts I want to introduce here. Number one is called the functional syncytium of the heart, or what that basically means is the cardiac tissue works together as this kind of interconnected network in this wave-like fashion. So if I draw a heart for you, that's a pretty good drawing for me. You can separate the heart into 4 chambers. And just to give you a little preview, the top chambers are called atria or atrium singular. So you have a right atrium and a left atrium. And then the bottom are called ventricles. You have a right ventricle and a left ventricle. Now how your heart actually beats is when the top layer, when the atria contract, the ventricles are relaxing. And then it flips. When the ventricles contract, the atria are relaxing. So the heart needs to be able to communicate with itself. It needs to be able to have all of the ventricle cells contract at the same time, but while shutting down the atria. And then when the atria contract, all the atria cells need to contract at the same time while the ventricles are resting. Otherwise, we're going to have blood going in weird different directions, not good for the heart. So how we do this is that branching and those intercalated discs. The intercalated discs, once again, allow those ions to sneak in and out of those tiny little spaces between the cells and allows the cells to communicate with one another and have a nice fluid, smooth electrical flow to it, and therefore the contractions are smooth as well. Now another cool feature of cardiac muscle is it is self-exciting, which means it doesn't always need to be stimulated by nerve impulses. So you see the cartoons or the movies where the person rips somebody's heart out of the chest and the heart's still beating. Can that happen? Yes, it can for a little bit, for like 30 to 60 seconds, and then it will die. So I don't want this slide to be misleading. It doesn't need stimulation by nerve impulses for a little bit. It can beat on its own, right? We have what we call pacemaker cells that help the heart maintain its rhythm. But please don't be misled. Your central nervous system still controls this. Specifically, it's something called the medulla oblongata. That is what's controlling your heart rate. So when your heart speeds up due to being scared, it has a lot to do with your central nervous system. If your heart beats faster because you're exercising, or if it beats slower because you're trying to go to sleep, that's all central nervous system input to the heart, and then the heart muscle cells just do what the central nervous system tells them to do. Okay, so yes, they can beat by themselves, but the brain is still in charge here, or the brain stem specifically. And another cool thing, one last note, is there is no tetany or fatigue with the heart. Thank goodness, right? Ever since we were born, our heart has hopefully been beating at a normal pace. And it's because, well, we'll talk about how the calcium and depolarization is a little different inside the heart, how the heart cells get excited. But the impulses don't occur rapidly enough to cause this. So the heart cells do get rest, right? As I mentioned, up at the top, once again, the atria, when they're contracting, the ventricles do get a little chill time. They do have to relax, and then vice versa. So the heart is able, even though it is beating every single second, there is a split little time where these cells can rest fully before getting stimulated once again. And then we move on to smooth muscle, and we've talked about this in the tissues chapter as well. Nice little review. Smooth muscle is found in the viscera, which basically just means your organs, specifically more hollow organs, right? So you see examples of the digestive system. So your esophagus, your stomach, your intestines are great examples. Your bladder has smooth muscle in it. Female's uterus, all your blood vessels, your arteries and veins, they all have smooth muscle inside of them. Your trachea, your windpipe, a lot of your airways, your bronchi and bronchioles, they all have smooth muscle. Anything that's hollow, pretty much smooth muscle. Controlling that. And similar to your cardiac muscle, it is involuntary. You cannot control your arteries and veins moving blood, right? Or the pressure. You cannot control your large intestine moving waste product. You have no say over that for the most part. All right, features of smooth muscle. There are no striations. That's why we call it smooth muscle. And when you looked at it under the microscope in chapter 4, what you should have seen was no stripes. So they are absent. Now, because there are no striations, because there's no altering myosin actin, dark light, there's really no Z discs. And if there's no Z discs, there's no sarcomeres. Okay, so how in the world are we going to contract muscle? The whole premise of this chapter was contracting muscle thanks to the sarcomeres, right? Well, yeah, but smooth muscle is just a little different. Smooth muscle still has thick and thin myofilaments. It still has actin and myosin, no big deal. But what happens is they're connected to the sarcolemma in this kind of crisscross looking fashion. And as you can see in the picture on the right, how this works is the muscle, when it contracts, it doesn't exactly get shorter from a Z-disc perspective, like a left to right. Kind of the whole thing just kind of scrunches up. So I picture like a sponge, right? You fill a sponge with water and then you just kind of wring the sponge and you kind of just bunch it all up in your hands. That's kind of how smooth muscle contracts. So it contracts from all possible angles and just kind of bunches or crunches up with that myosin-actin interaction. Other parts of smooth muscle is similar to cardiac. It is uninucleated, one cell, one nucleus. We do not have T-tubules in the smooth muscle. We do have a sarcoplasmic reticulum, but it's very, very poorly developed. For the most part, if you read most textbooks, it's pretty much non-functional. There is a function, but regarding what we've talked about with the calcium and the T-tubules, it really has nothing to do with that. So you can see in the picture on the right, I put a big red X covering all that, so it's pretty much, for our purposes, doesn't really do anything. Okay, poorly developed. Similar to cardiac muscle, the calcium comes from outside the cell. And we just have a different keyhole. Instead of troponin being the keyhole and calcium being the key, we have a different keyhole called calmodulin. So another big difference there. Troponin is found in your skeletal and cardiac, and calmodulin in smooth muscle. All right. Great chart. I would encourage you to either have this readily available or print this chart out or draw it on by yourself and review this. Make sure you know the differences between all three types of muscle tissues. And I'll go through it. I'm not going to go through it in great detail, but I'll just kind of skim through this with you. Do we have striations in cardiac tissue? Yes, we do. So I'll just put a check mark. We do not have it in smooth, and we do have it in skeletal. Okay. With those striations, they are interlinked with the sarcomeres. So we do have sarcomeres in cardiac. We don't have it in smooth, and we have it in skeletal. Number of nuclei. Remember, cardiac, any involuntary, is going to be uninucleated. So I'll just put 1x for 1 or uni. Cardiac and smooth are uninucleated, and skeletal muscle is multinucleated. So I'll just put, let's put 4 or more, right? There's a lot. All right, so I don't want to put 2x, meaning 2. There's a lot more than 2. It's multinucleated. Okay, with cardiac muscle, we do have T-tubules, and therefore we do have a sarcoplasmic reticulum, but it's not as developed. I'll put a check mark though, but it is there. It's just not as developed as skeletal. For smooth muscle purposes, no T-tubules, and the sarcoplasmic reticulum really doesn't do what we talked about in this chapter, so I'll put an X there. Skeletal, of course, T-tubules with the triad, and definite sarcoplasmic reticulum. With calcium, most of the calcium in your cardiac muscle comes from the outside of the cells. There is a tiny little bit inside the SR, but most of it's outside. Same thing with smooth. That calcium is going to bind to the calmodulin instead of troponin, and it's coming from the outside of the cell. Where in skeletal muscle, it's coming from the inside. Specifically, it's coming from the SR. Let's do that and be more specific, SR. And then control-wise, we know that cardiac muscle is involuntary, not in our control. Smooth muscle also involuntary. And skeletal muscle is voluntary. So make flashcards or study this table. Make sure you can do this table on your own from a blank slate, and you should understand and be good with all the different similarities and differences. All right, just a quick recap of some of the big things that we've talked about in the second half of this chapter, and we are good. So all review, no new information here. You can just kind of read this for yourself regarding all the things of the basic principles of muscle contraction. Remember, we talked about several different types of skeletal contraction. We talked about the graded strength principle and listening to certain music and arm wrestling a 5-year-old. We talked about a lot of weird things in this chapter, didn't we? Make sure that you are familiar with all of these bullet points. And then last but not least, on this slide, we're just defining a few of those types of contractions and principles, mainly on the bottom 2 bullet points from the previous page. Make sure you're familiar and good with these definitions, and let your instructor know if you have any questions. Study well, and I will see you for the next chapter.